Afternoon. Welcome to the afternoon session of the Markey Cancer Research Day event. It's really quite an honor to host this each year. And this year what we'd like to do is have Dr. Karf and Dr. Cassis come up and give us a few words regarding the amazing events that are ongoing in the Cancer Center, starting with Dr. Karf.
Thank you. As I think many of you know, I'm in the process of retiring. So I'm looking back at 14 years at UK Healthcare. And I must say, among the most special achievements for me over my 14 years is recruiting Mark Evers and helping create the Markey Cancer Center, the Markey Cancer Center Network, and the Research Network. I think through its research, it'll have impact on a national basis. But even more important, through its research, through its patient care, through its preventive medicine programs, and through the relationships with oncologists, it will change the care of cancer in Kentucky. It'll make cancer better, and it'll set a standard for cancer care in Kentucky. And I think the network has the opportunity to really develop a statewide standard, which will be a model for other cancer centers. So for me, I've seen this go from a couple of people to huge crowds. But I just want to say how very proud I am of what the Cancer Center has done over the, Mark, you've been here 7 years now, 8 years? 8 years. And actually, I'm anticipating even more once the submission goes in and gets reviewed. I think if Kentucky was fortunate enough to get a comprehensive cancer center, that would be an extraordinary achievement for the University of Kentucky, an extraordinary achievement the Markey Cancer Center, an extraordinary achievement for Mark Evers. Thank you for being here.
Well, good afternoon. It's great to be here. So, as your Vice President for Research, one of the great things I get to do is go across the campus and hear about research. And that includes having been present for many of the Cancer Center research presentations, interacting with you as groups or as individuals. And I have to tell you people, you really have your act together. You are committed and dedicated to what you do. It's so impressive to hear of your work. You're very well organized, and you're diverse, but yet you seem to be focused in ways that have made you nationally prominent. So I guess some of that certainly has to do with leadership. So I also want to thank Dr. Evers for all that he's done to promote cancer research and care here, especially through the NCI designation. And I wish him the best in everything that he has ahead of him and you have ahead of you. So I want you to know that the Office of Research is here to help you in any way we can. We celebrate your successes like today when you're sharing research and you have your distinguished guest with you. We also feel your frustrations, and we hope that we're not the cause of many of your frustrations. So to close out here, I wish you the best today. You cancer researchers, you keep going. We need you, and thank you for all that you do.
Each year for our program for the Markey Cancer Research Day, we pick two outstanding investigators to represent the depth and breadth of amazing work that's being done at the Cancer Center. For each of you that might have ideas about who you'd like to see next year, the Directors Council makes this decision, and so we vet that through them. So if you have any suggestions, please send them to me and I'll forward them on. This year, we have Dr. Robin Vanderpool. She's a native here of Kentucky, and she got her bachelor's degree of science in psychobiology at Center College, and then went on to get her master's in public health at Western College. She came to Kentucky in order to get her PhD, and she stayed on here as faculty and has been rising through the ranks ever since. She's a mainstay in the cancer prevention and cancer control and prevention program, where she's often seen leading very important initiatives in behavioral science. And with no further ado, Dr. Robin Vanderpool.
Thank you. All right. What an opportunity to present to my colleagues this afternoon. Thank you to Kathy, and thank you to the Markey leadership for, again, this opportunity. We're excited when CP can be focused on here at Research Day, and what I want to talk with you all about in particular is HPV vaccination and cancer prevention. And so I want to start with asking you a question. If there were a vaccine against cancer, would you get it for your children? All right. If there was a vaccine for breast cancer, would you not be shouting it from the rooftops? Right? Well, we have a vaccine that prevents cancer. In fact, we have a vaccine that prevents that could prevent almost 40,000 annual cases of cancer. But 10 years into this, now almost 11 years into this, we still haven't seen the great strides that we should in getting population level buy-in and uptake and completion of the HPV vaccine series. So that's what I want to talk to you all about today, and how the work that we're doing here at UK to help address this. So I'm going to do a very quick tutorial on human papillomavirus for those in the room who aren't as familiar with it. Where we're going to spend most of our time talking today would be this group on your left, those that are considered high-risk strains, those that cause cancer. And there's about 40 or so types that, again, affect the mucosal area. Types 6 and 11 are the ones that we commonly think of when thinking about genital warts and even low-grade cervical disease. But it's this group, particularly these 7 strains that are there in the red that lead to cancer. And 16 and 18 are two that we'll pay attention to as well. So some take-home points about HPV as a disease. It's the most common sexually transmitted infection. So there's problem one, right? We have to talk about sex if we're going to talk about HPV. And in the US, it's often controversial and taboo to talk about sex. As you can see, almost 80 million Americans currently are infected with some type of HPV. The good news is that 90% of the cases will go away on their own within 2 years, but what we are most concerned about is number 4 there, these persistent infections that may lead, again, to cancer and genital warts. And if you care nothing else, ignore the cancer burden, ignore the health outcomes that come from HPV vaccination. If you want to save some money, we should also invest in HPV vaccination. $8 billion annually in healthcare costs related to HPV-related disease. And this is the number that I gave you all earlier, and I apologize if this is hard to read, but these are some new data out of CDC that show that 40,000 cases, almost 40,000 cases each year, are linked to some strain of HPV. And this isn't one of those things where we think that this disease might cause, or this virus might cause cancer. We have data that shows that, again, almost 91% of all cervical cancer cases can be linked to an HPV type. Anal cancer, the numbers are as high as well, 91 and 93%, rectal cancer. And even our oropharynx cases of cancer, 70% can be linked to infection with HPV. And if you think about those that are directly covered by the new 9-valent vaccine that came out in 2014, you can see here that alone for cervical cancer, about 9,000 cases each year could be prevented by the new vaccine. Oropharynx cases, particularly among men, almost 9,000 cases could be potentially prevented by, again, population-level improvements in HPV vaccination initiation and completion of the full series. So let me ask you again, if there is a vaccine to prevent cancer, 40,000 cases each year, why are we not Why are we as a society not participating in this public health activity? And again, let's think about Kentucky. Let's think about where we live. If you look again at data, incidence rate data, you can see the southern part of the US, which covers a lot of other poor health behaviors as well and high rates of cancer. But for those cancers, again, associated with HPV, you can see that the higher incidence rates are here in the southern part, and particularly in Kentucky. And guess what? When I show you the map here in a few minutes of vaccination rates, they overlay pretty nicely. So again, I'm going to start kind of recalling some of the themes. We've had a vaccine for 10 years. It prevents cancer. You have to talk about sex in order to talk about this vaccine and also about the infection that causes it. Now let me add some more complexity to it. We have 3 different vaccines that are available. Originally, we started with the quadrivalent vaccine in 2006 that covered 4 strands, the 2 for genital warts, and then 16 and 18 that are linked primarily to the cancers that we talked about earlier. Then GlaxoSmithKline came along with a bivalent vaccine that only covers 16 and 18, mostly used in international settings, but again, still available. And then now, in 2014, we have a 3rd version of the vaccine, the 9-valent, which covers, again, the majority of the strains that lead to cancer or genital warts. So we have that has been part of our communication problem all along as well. And then even the recommendations continue to change over the past decade. We started with recommendations, you could start as young as 9 for girls and go all the way through age 26, but mostly focused on 11 and 12-year-olds, those adolescents. And as you can move across, boys were introduced here in 2009 as a permissive recommendation for vaccination. You could do it if you wanted to. And then in 2011, that's been changed to where males should be vaccinated as well. And then here we are toward the end of 2014 into 2015, where again, you can see the number of vaccines available for both males and females. And at this time, all of these are giving in a 3-dose series. So that's another complexity to this problem. You have a vaccine that you give at baseline, dose 1, and then you have dose 2, which is a couple months later, and then you have dose 3 that is 6 months later. So you have to keep up with all of that, right? And then just this past winter, we changed the recommendations again. Probably for the better, but again, another twist in the story of HPV vaccination, where it is now a 2-dose schedule, particularly those adolescents who are 9 to 14. So this is, again, relatively new from CDC. We, of course, haven't seen data reports come out with this yet because it just happened at the end of 2016. But as you can see here, if you're in that younger age group, now 2 doses. And you can get it at time point 0 and then anywhere from 6 to 12 months later. And then if you're older than that, the 15 to 26-year-old age group, you're still on the 3-dose series. So now we've taken the recommendations and divided them up by age, which has added another complexity of when you start recommending this to your patients and who gets what and when. So again, here we are 11 years later after the advent of the first vaccine, the quadrivalent vaccine, and only 6 out of 10 girls have initiated, that means just taken dose 1, and only 5 out of 10 boys have taken dose 1. And as I noted earlier, Kentucky falls in the category of having high HPV-associated cancers and lower vaccination rates. If you look at this in more specifics, you can see that, again, only about 1/3 or so of Kentucky girls have finished the vaccine series, and our boys, only about 17% have finished the series. And that is one of the lowest rates in the country. We're lower in national rates across the board as well. And then if you even think about the catch-up pool of individuals, those who are 19 to 26, we have even lower vaccination rates, but particularly for the males as well. So that's been part of the story and part of the complexity of HPV vaccination as well. We have gendered the vaccine, particularly in the early days of the marketing and the recommendations, that this is only for girls. And so we still have catch-up work to do in getting our males vaccinated as well. And what is particularly troublesome to me and others is that we can do better. We do better with other adolescent vaccines. If you put HPV vaccination in with the other adolescent platform vaccines that are given around 11 and 12, that middle school age period, particularly Tdap and meningococcal, you can actually get the same vaccination rates that we have for those two vaccines. But as you can see here on the the graph, we are much lower than our Healthy People 2020 goal of 80%, which we've already reached for these two vaccines, but we are well below that for males and females in HPV vaccination. All right. So I tend to give this presentation, and it's, Debbie Downer, and we have nowhere to go but up, hopefully, and that we have a lot of work to do. I still say that we have a lot of work to do, but there is starting to be some good news. Even with our low vaccination rates, there's three things that are kind of on the horizon here. With the two-dose coverage, the two-dose recommendation, we probably are going to see a bump, even in 2016 data that's going to come out later this year, later this summer, that we will see an increase in what you would call up-to-date, because at this point, then up-to-date is 2 doses, particularly for the younger age group. So is that because we're changing the definition? Or is it we're actually going to see increases in vaccination rates because it's easier to accomplish? So that's good news, that we'll have some more momentum to build on, hopefully, later in the summer. We're also starting to see decreases, a quite significant decrease in vaccine-type HPV infection among our teen girls. Now, if we had had the same coverage that countries like Australia and England had, we'd also be starting to see even more population-level impact on cervical disease outcomes. And in fact, we are starting to see some hints of that, very preliminary. In New Mexico, where they actually have an HPV PAP registry, they're starting to see decreases in all grades of CIN, particularly for the younger age group, and CIN2 for the older young adult women. And a lot of this is trying to figure out, are we starting to get cross-protection on some of the strains? Is less than 3 doses of the vaccine going to be okay? And, are we actually starting to get some effects of herd immunity in the population? So again, even though our rates aren't where they should be, and they're not at 80%, and we still have plenty of work to do, just think of the improvements on top of this that we could see if we reach those higher levels. So what happens in science and cancer research? In this case, we built a cancer prevention vaccine. So everyone's going to come knock on our door to get it, right? And in this case, science discovered HPV can cause cancer. A vaccine was developed. Dr. Lowy, director of NCI, was involved in those activities. Tested in clinical trials. Got it through the FDA and the immunization practices group at CDC who makes recommendations. So we're done, right? It's time to go. Everyone's going to want this vaccine. Everyone's going to come get this vaccine. All of our doctors are going to recommend it. All of our politicians are going to get behind it. We are ready. But that's, again, part of the story over 11 years of what's happened with HPV vaccination. None of this has come to fruition to the full impact. We've had to deal with social media. You can go on Snopes.com right now and type in HPV and get a whole list of myths that have been dispelled around the vaccine. There are dedicated Facebook pages against the vaccine. There's these breaking stories about investigators who developed the vaccine and can't sleep at night because the vaccine's so bad, that continue to circulate even today. Then you have celebrities who also are perpetuating myths about vaccine. You have presidential candidates who also on the campaign trail talk poorly about the vaccine and the, quote, dangers of it. And that is still talked about today. And you have a current president who also questions the science around vaccines. So none of this is helpful. Next, who would have thought that we'd have healthcare providers who would not recommend the vaccine? In some cases, again, whether it's personal beliefs, whether it's because they have to talk about sex, whether it's because they don't like confrontation with parents, we have instances where the vaccine is not consistently and strongly and frequently recommended by our healthcare providers. Then we also have this whole premise of fear of the vaccines among the general public. Again, we're 11 years into this, but people still think it's new. And then, even I have a story of a project we did down in eastern Kentucky where we were accused of giving the kids an HIV vaccine. And not in the preventive sense, but that we were giving them HIV. So you have to counter all of this in your work with HPV vaccination. And then this whole premise is, if I give my daughter or son the vaccine, it's licensed for them to go out and have sex, because they're protected now. And so we've had to work through, and all of this has been, particularly this last point has been disputed with data. Survey data, qualitative data, national surveys from CDC, all of this has been discounted. And we know with education and conversation and even training of healthcare providers, because they are one of the most important reasons why kids go on and get the vaccine and the parents agree to it, is the conversation with the healthcare provider. And then, what else? So, the community guide. How many in the room are familiar with the community guide? Public health people, come on. This is where we go to get evidence of how we can promote clinical interventions, for example, and in this case, vaccination. So here you can see 3 strategies for increasing vaccination. None of them, if any, have HPV vaccine as part of their evidence base. So we still really don't have a lot of evidence about what works to promote HPV vaccination, and mostly because they're small studies, like what I'm going to talk about today, that don't have a lot of population coverage. And then one would also think that there would have been a rush to help get this as part of the school entry vaccination requirements. Again, 11 years later, and we have 3 states, Virginia, Washington, D.C., and Rhode Island, who have school mandates for HPV vaccination. And essentially, if you sign a piece of paper that says you want to opt out, you can opt out. They're very loose opt-out. And, a state like Rhode Island is a lot different than a state like Kentucky. And the resources and their centralization of health care and public health services. So again, you can see all the dominoes and how they line up to impede our work with HPV vaccination. So with this, cancer researchers now have to think about partnering on many different levels. So if I was teaching this class in public health right now, everyone would know that this is a socio-ecological model that looks at the individual patient within a sphere of influence that goes all the way up to the health policy environment. And with this, you can see that if you're going to make an impact, you have to do multiple levels, you have to work with multiple groups, with multiple messages. And in this case, everyone from, again, working with payers and the Vaccine for Children's program, to working with school nurses, PTAs. How many cancer researchers work with their parent-teacher organizations? Outside of not being a mom, I'm looking at, and being actively involved with their children's program. And then working with immunization coalitions. That's been a switch, even at CDC and other groups, of having the cancer world talk to the immunization world and vice versa. Working within systems, in office clinics, and making an impact from everyone from the front desk all the way up through the providers. Working with pediatricians. Unless you're working in a pediatric oncology environment, you're probably not working with pediatricians on the downstream effects of not getting vaccinated as a child and the cancer outcomes many years later. And then, as I noted, culture, vaccine hesitancy, social media, all of that comes into play with the patients. So we definitely have our work cut out for us. A few years ago, the President's Cancer Panel took this initiative on, and as a result, the National Cancer Institute awarded supplements to 18 cancer centers to really get the lay of the land about HPV vaccination research, policy, media, education, provider work, everything that was going on in your state and your catchment area about HPV vaccination. And we were fortunate enough here at UK to be awarded one of those supplements. And our full report is on the Markey website if anyone's interested in reading it. We've published our methodology as well. And it really did set up opportunities to see where there's synergy between groups in the state, but then also where there are gaps in both research and practice. And I'll talk a little bit about that in a moment. As a part of these supplements, what was really groundbreaking is that all 69 cancer centers came together and have now released 2 consensus statements about the importance of HPV vaccination. So coming from cancer centers and their directors, all 69, and I know it's a little blurry, but there's the UK logo. We've now had 2 press releases about this, 2 consensus statements, and the last data that I saw, they had a reach of 146 million individuals from the last media push across the country for this. So again, getting our cancer centers more involved in this work. Quickly, I'm going to just run through some initiatives that we've done here at UK that I've collaborated with Dr. Alicia Cohen in communication, others in the College of Public Health. We've had funding from GlaxoSmithKline to, again, just change the conversation about HPV vaccination and this whole notion of, again, if there was something for breast cancer, we'd surely be taking care of this. And that it does have to relate to sex, and we can talk about it. It's okay. Another push that's been going on both here in Kentucky and nationally is getting survivors involved, because they have very powerful stories to tell, because they have cancers and gone through that experience, that disease can now be prevented. No one else has to go through that. And so we've had women here in Kentucky share their stories, and then nationally CDC and the Head and Neck Cancer Alliance also now has these stories. And many of the cancer centers have gotten involved in getting their survivors as part of this. Another is framing this in ways that the youth understand, right? So having HPV, you are part of a social network, right? 79 million people are part of that social network. And so sharing it's light and funny, but serious at the same time, of talking about this and how you, in this case, don't want to be part of a social network. We've been working in the schools. Again, you've got a captive audience. You need a school system that's supportive. And in this case, we were able to collaborate with Lake Cumberland District Health Department and their school nurses. And we delivered an HPV vaccination intervention where those students who started in August, 88% of them completed the 3-dose series by the end of the school year. The parents didn't have to take them out of school. We got permission to give the vaccine in the schools by the school nurses. We gave the vaccine away for free. We made it part of the social norm. We got the kids involved. How many of you all remember the old MTV logo? The kids designed that poster for HPV and making sure that others in the school were vaccinated. And again, and this is just another way of showing also community engagement for the Cancer Center and for the university of partnering with those in practice. To have access to 900 kids in a school system is nearly impossible if you don't have the buy-in from those schools and the administrators. Another intervention that we've done quickly is 1-2-3 PAP. This is where we focused on the catch-up population of young women in eastern Kentucky. Again, strong component of community engagement where we actually gave out dose 1 for free, and the whole point was to then randomize them into a study to help them with adherence to dose 2 and 3. Because that's another issue which we don't have time to talk about today, is adherence to the vaccine schedule. It again adds that other layer of complexity. And with this, we then took members of the community and had them star in our video about HPV vaccination and adherence and eliminating barriers. And these are all people from the community that they recognize, including 2 healthcare providers and a local TV personality. So using the people from the community to deliver the public health message and program. Long story short, it was successful. Those who got our intervention were more likely to complete the 3-dose series. Of particular importance for cancer prevention and control is dissemination of these effective evidence-based programs. And we've now reshot the video for the state of Kentucky, North Carolina, and West Virginia. But back to what I was saying earlier, due to the changing vaccination guidelines, we would have to almost start from scratch to redo this for the new 2-dose series and also incorporation of males into this. But that being said, you know, we have had success with this and designing our programs from the very beginning so that others can adopt them in the future. And we are also, even the National Cancer Institute, there were no evidence-based interventions on their Cancer Control Planet website for HPV vaccination. We were the very first one. And there's only been 2 since. So again, much more work to be done. And then lastly is one of our partners in the environmental scan that I talked about earlier. We identified pharmacies as a potential opportunity, right? How many of you all have been to a pharmacy this week for something? See, quite a few hands. More than probably have been to the doctor this week. And so we know that pharmacies can give pneumonia vaccines, influenza vaccines, and even shingles vaccines. So why not see if HPV can work there, particularly because of the later hours, the more convenient schedules. And so we partnered with Total Care Pharmacy in Moorhead to implement a protocol that the Gateway District Health Department served as the medical supervisor for. We did continuing education through our AHEC program and really covered a lot of media and promotion around the availability at the pharmacy. Now, I could spend a whole presentation on the barriers and lessons learned from this project, but I think until we see greater policy support for HPV vaccination, particularly for, say, school entry, we're going to have to do these small pilot type of projects that we can make improvements on. And in fact, if we did this project we just did it last summer. If we did it again right now, there's even been a new policy change at the state that pharmacists can now vaccinate down to age 9 without a medical prescription from a physician. So again, I think there's lots of lessons learned about how we can increase access to HPV vaccination. So in the sake of time, I just wanted to say we've got more things to come, so hopefully I'll be invited back in a few years to give you some updates. We have another administrative supplement from NCI where we're looking at our catchment area and their health behaviors, and we're going to be able to dive down and really focus on HPV vaccination in one of the set of questions that we have. Several of us are collaborating on a PO1 application that'll go in the fall. And one of the projects is on HPV vaccination in a clinic environment. Again, addressing everyone from the patient all the way through up the office staff and providers. We've been doing some work around community clinical linkages. That's a very hot topic for CDC. What does it take when you put clinical entities that can deliver the vaccine partnered up with their community organizations? And then just so it's on everyone's radar, NCI is very, very interested in rural cancer control issues right now. There'll be some funding announcements, I think, in the next year or so focused on that. So again, we want to turn our attention to how we can to bring HPV vaccination into that. And then also work with our federally qualified health centers across 3 different states. Again, an underserved population that we can increase HPV vaccination within a clinic environment. So with that, I will stop and say thank you to all of my colleagues and collaborators and funding sources for the work that we've done, and I'm happy to take any questions that you have.
I do ask that you use the microphones for any questions, because we do have a live feed going to our affiliate network for these questions. Thank you.
Hi, very nice update.
Thank you.
Why does recommendations end in the mid-20s Aren't older people susceptible to this?
Right, right. So from the best of my knowledge, and others in the room may be able to speak to this better, is, you know, the immunogenicity data is based on that age range of up to 26 and the impact that they've seen over the long term of being able to sustain protection against the virus. But you're right, we are now seeing, when I was in South Carolina last week for the Hpv vaccination meeting, a GYN oncologist was sharing that she had an older couple in her practice that they've been dating, and they're in their 60s, and they came in and got the Hpv vaccination. Of course, they had to pay for it out of pocket, but you know, there is you know, the possibility to still transmit HPV even in older populations, but the vaccine requirements still haven't been updated for that. Yeah.
Hi, I was just curious about the refusal rates. So for the Lake Cumberland project.
Oh, sorry, I couldn't see where you were.
More on the side. So the 88% that completed the series is great. For the 12% who didn't finish, do you have a sense of why there was failure for the completion of the series? And then what the initial refusal rate was like among the 900 sort of population that you had?
Yeah, so there's a whole presentation to be given on that project as well, and I'd love to talk with you about it. For those who didn't finish the series, a lot of it was just some of the kids, they move out of the schools a lot. The mom that I told you said that we gave her child the HIV vaccine, pulled that student out and did not let them finish the series. We had a couple pregnancy cases. So some just, you know, kind of quirky things that happen with some of that group. And then I don't have the data in front of me, but we did struggle with getting permission in the first place. You know, the back to school packets that their kids get in their backpacks, the HPV vaccination had to be on a standalone form versus kind of incorporated with the rest of the vaccine pages and school nurse information. And so I think there was some folks who just ignored that altogether and never returned it. And then we had some kids who had even started the vaccine, very few, but we had some that had started the vaccine series elsewhere or even completed it elsewhere. So the ends move around quite a bit, so I tried to get to the take-home point, but I'd love to share that paper with you.
In relation to low rate of the completion of vaccine procedure, I'm wondering what is the side effect of vaccine, what is the usual cost of vaccine?
The side effects, is that what I heard? So they're really no different than the majority of other vaccines where some get a red, some of you all in this room have probably had it, I'm too old, that it does hurt. But I would argue many people will say shots hurt. And, you know, potentially the redness or even swelling of the injection site. And there's been some cases of individuals passing out, syncope from the vaccine. But I think that also holds true for many other adolescent shots. But there have been they've been monitoring the vaccine data, the side effect and adverse events data for 11 years now, and it's no different profile than other vaccines.
Okay, thank you for all the wonderful questions. We can continue the questions at the reception.
Yeah, thank you.
I would like to I hate to end a good discussion, but what are you going to do? So our next speaker is Dr. Tianyan Gao. I've known Tianyan for actually quite a long time now because she was recruited to UTMB when I was there and moved with me and is actually now my next door neighbor in the BBSRB building. So Tianyan was originally from Shanghai, China, and came to the United States to get her PhD at Northwestern University. She then had the honor to go to University of California in San Diego to work with Alexander Newton, who's actually quite famous in the field of signal transduction, where she was able to discover a protein called FLIP, where FLIP is the phosphatase that inactivates the AKT pathway. And today, I'd like to have Dr. Tianyan talk about her work.
Thanks everybody for being here. Thanks Robin for the wonderful talk. It's really hard to follow her lead. Many of you know my lab has been working on a protein phosphatase called PHLP for more than 10 years now. And this phosphatase is, oh, I totally forgot. This phosphatase was initially identified when I was postdoc in Dr. Alexander Newton's lab when we were working on protein kinases. And then our keynote speaker, Dr. Tony Hunter here, was among the first critics and supporters of our studies. And so it's especially, I really especially appreciate this opportunity to share some later studies from my lab and also our progress made on determining the tumor suppressor function of PHLP in colon cancer. PHLP stands for pH domain leucine rich repeats protein phosphatase. It's a small family of protein phosphatase, have two members and have two genes, PHLP1 and PHLP2. And PHLP1 has a potential alternative splice variants to give it two different isoforms. And PHLP is evolutionarily conserved. The earliest isoform of PHLP homologs we can trace back is from Drosophila. And I'm actually going to show you some data we initially generated back at UCSD. And PHLP, as indicated by its name, it contains multiple regulatory modules in addition to its catalytic domain. So both of the PHLP isoforms contain these phosphatase domain, similar to PP2C-like phosphatase. And in addition to that, it has other regulatory domains, for example, pH domains and leucine-rich repeats added to the protein. And at the end of the phosphatase, both of the phosphatase has these PDC domain ligands to allow them to bind PDC domain-containing proteins. So this is actually a newer structure models generated with help from a crystallographer from my department, Dr. Konstantin Korotov. And both of the phosphatases share this conserved LR motif, form this horseshoe structure, allow this domain potentially to bind other regulatory proteins. And both of the phosphate domains from PHLP1 and PHLP2 have this conserved PP2C-like structure with all the residues I labeled here are required for coordinating metal ions for catalysis. And so before we hear about Dr. Tony Hunter talking more about kinases, I'd like to borrow some of the numbers from his seminal work on defining the kinase families. So I just want to give you a rough idea of how phosphatases compare to kinases in terms of number in the human genome. Human genome contains a large number of protein kinases. It's over 500 of them. And among the kinases, the serine threonine kinases are the major kinase family in the human genome. It's close to 200 of them. Compared to the protein kinases, the protein phosphatase family is considerably smaller. It only has less than 150 members. And importantly, among the members of phosphatases, you can see the tyrosine phosphatases actually have relatively similar numbers compared to the tyrosine kinases. Surprisingly, with such large number of protein kinases, of serine protein kinases, there are less than 40 of serine phosphatases actually exist in the human genome. And they're subdivided into additional families based on the mechanism of catalysis. The PPP family has 13 members, and these are the family are better studied and has some of the phosphatases that you probably know in your own studies, such as PP1, PP2A, and calcineurin. And the PPM family contains PP2C and different members of PP2C family, and PHLP belongs to this PPM family. As already alluded to, the phosphatase activity of PHLP requires a coordinating of metal ions such as magnesium and manganese. and there's other type of aspartic-based phosphatase as well. In earlier studies, when their proteomic analysis of mammalian cell, and in the study they were characterizing all kinds of different phosphorpeptide from a human cell, and among more than 6,600 phosphorylation sites, over close to 98% of the phosphorylation events actually occurs on serine or threonine residues. There are only about 2% of the phosphorylation events actually occur on tyrosine residues. So as you can see, with such a small number of phosphatase around in the cells, and they have to do this huge job to control the phosphorylation of many different phosphorylation events occurring inside the cells. So it's not surprising each of the serine threonine phosphatases actually are involved in dephosphorating multiple different substrates and involving in controlling a variety of cell signaling. And this cartoon depicts, is published back in 1997. At the time, people just the family of phosphatases had started expanding. People start to realize there are more phosphatase around in the cells. However, the scientists were struck by the fact the phosphatase actually share very little conservation at the protein level, and that made the study of the phosphatase actually more challenging. And the fact they are more eccentric proteins make the studies actually more interesting and also sometimes somewhat difficult. And our studies on PHLP as a protein phosphatase actually stem from our study back in my postdoc lab, where we are initially most interested in study protein kinases and how phosphorylation regulates protein kinases activation. As most of you know, one of the protein kinases we studied in the lab was AKT, and activation process of AKT requires this two-step of phosphorylation. AKT is activated downstream of PI3K and downstream of receptor tyrosine kinases, and production of the second messenger PIP3 molecule at the membrane allow AKT to translocate to the plasma membrane, where it can be phosphorylated by two different upstream kinases, PDK1 and mTOR in the TOR2 complex. And once phosphorylated, AKT can go into different locations of the cell, and downstream signaling from AKT can lead to oncogenic transformation of the cells. This pathway is known to be inactivated by a lipid phosphatase P10, a well-known tumor suppressor in many different cancers, and P10 by removing activation signal, dephosphorating PIP3, prevent AKT activation. At the time we were looking at this signaling pathway, we were searching for some other inactivating molecules of this pathway. Identification of PHLP actually is based on a simple, somewhat naive hypothesis, is because activation of AKT requires a pH domain of AKT leads to this membrane translocation step. We are thinking maybe there was a phosphatase exists in the cells as well. Having a pH domain coupled to a protein phosphatase domain may allow this phosphatase to specifically regulate this pathway. We are searching into the database. We're actually quite lucky to identify there are only two phosphatases fit this criteria. Those are the FLT1 and FLT2 genes. Later on, the pH domain of PHLP didn't turn out to be the critical requirement for dephosphorating AKT, but sometimes science needs luck as well. I want to share with you, so this is really old data from my initial identification of PHLP as a phosphatase of AKT. And in this simple experiment, we used 293 cells and overexpress of HA tag FLP1 into the cells, as overexpression of phosphatase leads to decreased activation of AKT and shorten the activation time course stimulated upon insulin activation. And then, since AKT pathway is highly conserved from Drosophila all the way to human, we reason if FLIP is an important phosphatase for regulating AKT, and there may be the phosphatase back into, their phosphatase similar to FLIP exists actually in Drosophila and can serve as the same function. It's actually indeed the case. I was actually really proud of this study at the time because we had no idea how to knock down FLIP, knock down Drosophila isoform of FLIP in the Drosophila S2 cells, where with the help from Jack Dixon's lab, we actually did it. We, as in this example, in insulin-induced activation of AKT, when you knock down PTEN, the activation of AKT is significantly elevated, and similar effect of AKT activation can be achieved if you knock down the Drosophila isoform of FLIP. So that gave us a confidence that FLIP indeed serve as an important signaling molecule to inactive AKT, can achieve a similar function as a PTEN. So when I started my own lab, and so when we want to use a cancer model, which we picked colon cancer as our model by collaborating with Dr. Mark Evers. And the lab is basically working on, and there are two overarching hypotheses or overarching goals. First is we like to understand better of how FLIP phosphatase is regulated by upstream regulators, and the fact that each of the protein phosphatase is likely to control multiple different substrates and effectors downstream. So we also interested in identify additional downstream effectors of FLIP. I still remember back in San Diego when I first presented our data on FLIP as a protein phosphate for AKT in one of the departmental talks for a postdoc, and Dr. Tony Hunter was there, and afterwards, in my summary statement, I started to suggest FLIP may be a tumor suppressor because they can dephosphorate AKT, and later Dr. Hunter talked to me, and Alexandra said, if you want to say something is actually a gene is a tumor suppressor, you actually need to find direct evidence these genes can actually have some mutations or alterations in the human cancer cases before you can actually say it's tumor suppressor. So one of our goals initially when I started to working with FLIP in colon cancer, we actually do want to find evidence that FLIP can be altered, the expression can be altered in colon cancer as well. And after the database from Human Cancer Atlas project becomes available, by collaborating with the bioinformatics, biostatistics core, we were able to show indeed both FLIP1 and FLIP2 genes are significantly downregulated in colon cancer specimens compared to normal controls. And there are actually point mutations can be found in FLIP1 and FLIP2 in colon cancer as well. Some of the mutations actually are quite obvious. They have truncation frame shift or premature codons in the middle of the protein that leads to deletion of the phosphate domain. Some other mutations we have not yet to figure out the functional impact of those mutations are. Following our studies and other people's have also provide evidence that loss of FLIP expression is actually commonly associated with other cancer types as well. In this study, the authors show the copy number loss of FLIP1 and FLIP2 genes actually occurs in large percentage of prostate cancer patients as well, and especially in the metastatic prostate cancer, loss of both FLIP and PTEN seems to promote the metastasis and tumor progression of prostate cancer. And there are also other ways to lose FLIP gene expression, and one of them in this paper, people show in melanoma, FLIP can be suppressed by hypermethylation of the DNA. And there are a number of studies later came out to show both FLIP1 and FLIP2 genes are targets of oncogenic microRNAs or SNO RNAs. And in many different cases, these oncogenic microRNAs can downregulate FLIP expression directly. And so in my lab, we also try to find additional mechanisms that can lead to downregulation of FLIP in colon cancer. One of the mechanism we focused on was proteasome-mediated protein degradation. In here, I only want to summarize a couple of studies from the lab in 2 slides. One is showing we identify a E3 ligase, beta TRCP, as a E3 ligase to leads to ubiquination of FLIP. In here, as you see, overexpression of this E3 ligase promote ubiquination of FLIP protein and leads to degradation of FLIP. And these are the substrate binding mutants of beta TRCP lacks this function. And later on, we also identified the reverse reaction. The enzyme responsible for deubiquinate FLIP is USP46. And in similar experiment, when you overexpress USP46, it actually leads to deubiquination of FLIP1, and then the enzyme dead, deubiquinase does not have this function. And so and this is actually relevant to tumorigenesis as well. So in this study, we showed hypoxia, which is commonly associated with many different types of solid tumors. In this case, we show in the colon cancer cells, if you subject cells to hypoxia with 1% oxygen, you induce degradation or decrease expression of both FLIP1 and FLIP2 proteins. And it also leads to decrease of USP46 expression in the cells under hypoxia, and that is, the mechanism is hypoxia leads to mRNA downregulation of USP46, and that's the mechanism leading to FLIP downregulation in hypoxia, and could contribute to the tumorigenesis process. And if you actually gave cells back deubiquinase to prevent the degradation of FLIP, the stabilization of FLIP enhances its ability to inhibit tumorigenesis. Here we either express the FLIP1 by itself or FLIP1 together with the deubiquinase USP46. You see a combination of the two together leads to a much more significant dephosphorylation of AKT, and that also translates to decrease the tumorigenesis in vivo using xenograft mouse models. And you can see these, there's almost complete inhibition of tumor growth where the control cells have a much higher tumor volume. And so to summarize some of our studies on studied regulation of FLIP expression, in the lab we were able to show FLIP is phosphorylated downstream of a couple of kinases, and this phosphorylation leads to beta TRCP dependent ubiquination and degradation of FLIP, and this process can be reversed by a deubiquinase USP46. And in the cases of tumorigenesis, and this process can contribute to tumorigenesis by at different levels. The E3 ligase beta TRCP has been shown to be downstream targets of Wnt and beta-catenin pathway, which is commonly activated in colon cancer. And we show hypoxia can inhibit USP46 expression, so destabilize FLIP and decrease FLIP expression. And we also, later studies, we found chronic inflammation as associated with IBD patients, and those patients are known to have increased cancer risk. And in these cases, the inflammation can promote FLIP degradation by the proteasome pathway. So all of these mechanisms can lead to FLIP downregulation and promote tumorigenesis. But today, I want to actually talk about it a little more beyond that. So in our initial studies, we found FLIP can antagonize AKT pathway and downstream mTOR as well, and that can leads to inhibition of cell proliferation and promotes apoptosis. And these we show both using in cells and also using xenograft mouse models. Some of our later studies, we found FLIP can also control the RAS-RAF pathway downstream of RTKs, and dephosphorylation of RAF1 mediated by FLIP can inactivate RAS-RAF pathway as well. We show using a FLIP1 knockout mouse model. We show if you knock out FLIP in the APC mean mice, you induce these invasive growth of tumor cells, invaded into local mucosal layers, and that is because the mechanism is the FLIP can inhibit tumor progression by antagonize RAS-RAF signaling. And one of the most recent studies, we also show FLIP can inhibit atypical PKCs, and that controls the cell polarity in epithelial cells. In the relatively normal epithelial cells, you can form these nice polarized structures, as the patent shows you earlier today. And if you knock down FLIP, you get this multi-lumen formation of in the 3D structure, and you also promote the proliferation and anti-apoptosis effect when you don't have FLIP. And this effect can be mediated by FLIP-mediated regulation of atypical PKC activity. In the next few minutes, I want to actually share with you a new studies we started in the lab, try to look at FLIP function in regulating cellular metabolism. And so many studies before, we all know, it's well known, AKT, it plays important role in regulating glucose metabolism, both in insulin, in normal tissues, in insulin responsive tissues such as muscle and heart and the brain, but also the AKT is also known to promote glycolysis in cancer cells as well. So the glycolysis is once the glucose get into cells, undergo multiple different steps of glycolysis, and production of pyruvate can go into the TCA cycle in the mitochondria, where it can contribute to ATP production, leading to regulating the bioenergetics, and also the intermediate metabolites formed inside the mitochondria contribute to biosynthesis of other molecules in the cells to allow cancer cells to proliferate. And more recently, these mitochondria has been shown to play an important role in regulating cell signaling as well. If you have increase of glycolysis in the cell, sometimes in addition to the pyruvate going to the mitochondria, it also leads to a production of lactate and the secretion of lactate into the cell medium. So when we were study FLIP, we actually observed this really interesting phenotype. The FLIP knockdown cells always seems to, the growth medium always seems to turn yellow very quickly. So that's at the time we were thinking maybe FLIP can regulate these glycolysis pathways, somehow change the lactate production in the cells. And so we started to look into whether FLIP plays a role in regulating cellular metabolism. And indeed, when we were measuring when we measured the glucose uptake and lactate production, which are the indicators of glycolysis capacity in the cells, we see these knockdown either FLIP1 or FLIP2 have to allow cells to increase glucose consumption in two different kind of colon cancer cell lines, and also that coupled with increased lactate production. And when we measure these cellular metabolism more specifically using seahorse, we were able to see when glucose induced the extracellular acidification rate, which indicating how cell you produce lactate. And when you knock down FLIP1 and FLIP2, and these at multiple different stages, you had increase of the ECAR production, ECAR rate. And if you quantify these, knockdown FLIP1 or FLIP2 is linked, results in increase of glycolysis and increase of glycolytic capacity, but not on the glycolytic reserves. And we also measured the mitochondrial activity specifically in this case by measuring OCAR, oxygen consumption rate. And in these cells, again, knockdown FLIP increased both basal respiration, showing here, and maximal respiration, this step, and also the respiration correlated with ATP production. So sometimes when the cells switch between mitochondrial respiration and glycolysis, they will prefer one over the other. But in the case of FLIP knockdown, cells to generally change their metabolic behavior to have both a reduction of glycolysis and oxidation capacity, as shown in these metabolic phenotype chart, you see on this axis, if the ECAR rate is more towards the right, meaning cells are more glycolytic, and also when the OCAR levels are higher in the FLIP1 and FLIP2 knockdown cells, this indicates knockdown of FLIP1 and 2 make the cells more glycolytic and also have increased the mitochondrial respiration. So this suggests to us maybe FLIP is regulating some steps upstream of both the more upstream of in the glucose metabolism utilization pathway. And when we are looking into the molecular mechanism, we found knockdown either FLIP1 or FLIP2 leads to increased phosphorylation of HK2, hexokinase 2, which is the first key enzymes control glycolysis to allow the glucose committed into the glycolysis pathway, and also in as control, we also see in both FLIP1 and FLIP2 knockdown cells, AKT phosphorylation is also increased. And this is reversed when you have overexpression of either of the FLIP1 or FLIP2 phosphatase. You have this decrease of HK2 phosphorylation and decrease of AKT phosphorylation. And so since the HK2 is also known to partition either in the cytosol or in the mitochondria, or can be associated with mitochondrial membrane, and association with mitochondrial membrane of HK2 actually indicates a higher contribution of HK2 in regulating glycolysis and also regulating mitochondrial metabolism. So we in here we analyze the cellular fractionation and cellular localization of HK2 in the control and FLIP knockdown cells, and you can see HK2 when you fraction the cells into cytosolic fraction or mitochondrial fraction, you can see in the FLIP1 and FLIP2 knockdown cells, you have increase of HK2 partitioned into the mitochondrial fraction, and this can be quantified in this bar graph showing the significant increase of how much HK2 is actually associated with mitochondria, and that also associated with a higher mitochondria associated HK activity as well. So all of these suggest loss of FLIP can promote mitochondrial association of HK2 and increase how much hexokinase activity at mitochondria. And this is also interesting to us because we are biochemists by training. We also want to see if some protein phosphatase can interact with something to achieve its activity. In this case, we again fractionate cells into cytosol and mitochondria fraction, and then did the immunoprecipitation with HK2 antibody. As you can see, HK2 can be precipitated from both cytosol and the membrane fraction. But interestingly, only in the mitochondrial fraction, you actually see these both AKT and FLIP1 and FLIP2 proteins can form a functional complex with HK2. This is how much protein being inputted in the IP. Even though the HK2 is a minor fraction of HK2 is actually associated with mitochondria, however, that fraction of HK2 can form a complex with both the kinase and the phosphatase in the same signaling complex. We're initially quite excited to test whether HK2 can actually is a direct phosphatase substrate of the FLIP1 or FLIP2, but it turned out we cannot rule out the possibility that FLIP1 or FLIP2 regulate HK2 through AKT. So in this experiment, we did to tease out this interaction a little bit more. We used humanized T cells overexpressing HK2 and with FLIP either in absence or present of AKT, and test to see how this complex formation can be regulated. And we, as you can see here, only when we have overexpression of AKT and this complex can actually form, this stable complex can actually form with both, in the complex has both the, has all the HK2, AKT, and the FLIP. But without AKT, this complex actually did not really form together. So this is suggesting to us the FLIP can control HK2 phosphorylation, membrane localization of HK2, but mostly likely goes through AKT. So to further test if HK2 actually mediates some of the glycolytic effect we see in the FLIP knockdown cells, we did this experiment to use a compound clotrimazole. This is antifungal drug. Recently has been identified to have some anti-glycolytic glycolysis activity by dissociating HK2 from mitochondrial membrane. So in this experiment, we test if we just simply dissociate HK2 from the mitochondrial membrane, whether that is sufficient to block any of the FLIP loss-mediated effect on glycolysis. So in here, I'm just showing you the compound actually works of dissociating HK2 from mitochondria. HK2 basally associated with mitochondria, and in the FLIP 1 and 2 knockdown cell, you have more association with mitochondria. When you treat with this compound, you dissociate HK2 from the mitochondria fraction altogether, both in the control and knockdown cells. And indeed, when we do that, we will actually be able to block FLIP-mediated increase of glycolysis. As you see here, in the without this compound treatment, FLIP1 and FLIP2 knockdown cells leads to increased glucose consumption, increased lactate production, indicating increased glycolysis. But if you treat with this compound, this increase is totally abolished. And finally, we did this experiment to directly knock down HK2 to show the FLIP actually functional using HK2 as the mediator to control glycolysis. So in this side of experiment, we knock down HK2 in the control or FLIP knockdown cells. You see the HK2 knockdown in these set of cells, and FLIP knockdown showing over here. And the knockdown HK2 did not change the other isoform of HK1 expression at all. And in the knockdown cells of FLIP1 and FLIP2, you see this basal increase of glucose consumption and lactate production, indicating increased glycolysis. However, this increase was abolished, mostly abolished when you knock down HK2 from these cells together. And finally, the functional implication of our results is we reasoned that if loss of FLIP knockdown made the cells more rely on glycolysis, the cell upregulate glycolysis as a way to survive, then these cells will become more sensitive when you actually inhibit glycolysis. So this turned out to be the case. In a couple of different colon cancer lines, we starve the cell, or we treated cells with 2-deoxyglucose, the 2-DG, to inhibit glycolysis. The 2-DG competes for normal glucose to enter the glycolysis cycle, but block that glycolysis once it becomes phosphorylated. And in the cells treated with 2-DG, the control cells, you inhibit some of the cell growth. You have about 75% of cells survive. But then the FLIP1 and FLIP2 knockdown cells, they become more sensitive to 2-DG-induced cell death. So you actually have more cells, less cell survival in both cases. This is actually really interesting. In the basal medium condition, FLIP knockdown cells actually make the cells survive a little bit better. But if you starve cells specifically of glucose and by inhibiting glycolysis, these cells become more sensitive to glucose deprivation-induced cell death. So to summarize this part of study together, we showed FLIP can directly control the phosphorylation of HK2. Phosphorylated HK2 become associated with mitochondria, where they can promote the glycolysis and also promote the TCA cycle of mitochondrial activity. By having FLIP to block the phosphorylation, FLIP can inhibit the glycolysis process to inhibit additional mitochondria-mediated advantage to the cancer cells. And finally, to put all of our studies together, the effort from the lab in the over the past 10 years have identified a number of upstream regulators of FLIP, including beta-TRCP, USP46, I talked to you a little bit about. And we also identify a scaffolding proteins can actually bring FLIP to the membrane to antagonize AKT. And FLIP is also a translation targets of mTOR. And downstream of FLIP, there are, in addition to AKT, we identify additional downstream targets of FLIP, including RAF1, SSK, PKC isoform, and also the HK2. And together, having FLIP as a tumor suppressor exists in the cells, it can function to inhibit cell growth and proliferation and induce apoptosis, and also inhibit the migration and metastasis in the tumor progression. And finally, we show FLIP actually controls cellular metabolism as well. Many people contribute to our work, and our current lab members, so we have 2 postdocs and Peyton as a graduate student. All of them work really nicely. We work nicely together. We try to make more progress, hopefully. And we have a visiting scholar in the lab now, and also have undergrads funded by the Markey Cancer Center fellowship for undergrads who wants to pursue oncology in their future studies. And there are many formal members of the lab contributed greatly to our study and to some of the earliest data I showed. And we have wonderful collaborators, our longtime collaborator, Dr. Mark Evers, and our biostatistician, Heidi Weiss, and Chi Wang, and pathologist, Ang Lee. And we also recently started a collaboration with Dr. Terry Barris. And the choreographer in my department, Constantine, helped me with model the structure of FLIP, LR, and phosphate domain. And at UCSD, my former mentor, Dr. Alexander Newton, has been continuing collaborating with us. And the HK2 study was supported by my friend and colleague at UCSD, Shigaki Mariotto, for the HK2 construct. And our studies are supported by multiple different Markey Center shared resource facilities, and also fortunately funded by different funding sources. Thank you for your time.
We can entertain 2 quick questions.
And you show that the tumor cell, which has upregulated FLIP, has very suppressed growth in vivo when they injected it. So I'm wondering that, have you ever do the challenge injection of the wild type tumor, whether that those that injected animal become resistant to the challenge of the wild type cancer?
So you're asking about the tumor genesis experiment when FLIP was overexpressed?
No, that you show that the tumor cell, which is induced upregulation of the FLIP, doesn't grow very well in vivo.
Right.
So in those mice, have you ever do the challenge injection of wild type tumor?
So the only tumor genesis experiment we tried is to make the stable cells where you overexpress FLIP, and that caused the decrease of tumor growth, was indicating a tumor suppressor role. We haven't tried to manipulate FLIP expression any other way.
Yeah, the reason why that I ask is that maybe that those that genetically modified tumor cell can be the very good inducer of protective immunity. It's kind of long shot, but if it does, it's very useful too.
Right. Thank you.
In terms of therapeutic implication, do you have any small molecule that can promote overexpression of FLIP, or on the other hand, do you have a target that you could inhibit, and by doing this will overexpress the activation of FLIP? So the question is, do you have a small molecule that can activate FLIP?
Okay. No.
Okay. Now, now the next question is, if you had a target that you'll select, that we could inhibit, so that by doing that, we could control FLIP.
Yeah, so that's the challenge of working with tumor suppressor. Usually once it gets lost in cancer during the tumor genesis and progression, it's really hard to get it back. And I guess if you manipulate the ubiquitination pathway, potentially you can increase some of the tumor suppressor expression.
Okay,
Thank
that
you.
will conclude our faculty talks. We will take a very brief 5-minute break, and then we'll have the State of the Cancer Center address by Dr. Evers. May I have your attention, please? We'll go ahead and continue our program. It is my great honor to introduce Sally Humphreys, the president of the Markey Cancer Foundation.
Thank you, Kathy. Good afternoon. Our foundation's mission to support innovative cancer research and treatments guides us in all that we do. And today's program is an excellent example of that fulfillment, joining you in looking toward tomorrow and the discoveries the new day may bring. We're proud to support your work as you demonstrate such an important aspect of the human spirit, the drive to explore and to discover. Most importantly, you do this not for personal gain, but to alleviate suffering and improve the human condition. For this, we are thankful, and of this, you should be proud. It is now my honor to introduce to you the remarkable Dr. Mark Evers. Mark was voted one of the best doctors in America 2016, an honor only 5% of doctors in America earn. It is bestowed by an impartial peer review by Best Doctors, Inc. of more than 40,000 physicians. Dr. Evers, a surgeon scientist, is the director of the Markey Cancer Center. Additionally, he is the physician and chief of the oncology service line, a full professor and vice chair for research in the Department of Surgery. In 2010, he was named winner of the American Surgical Association's Flance Carl Award, the highest research award bestowed upon an American surgeon. Mark led Markey to NCI designation in 2013 and will soon lead Markey to comprehensive status in 2018. It is such a privilege to work with Mark, and it is our honor to support his vision. Ladies and gentlemen, my hero, Dr. Evers.
Okay, thank you all very much. I really appreciate everybody coming. This is another great event. I think this is our 8th event, our 8th Markey Cancer Center Research Day. Seems to be getting better and growing more every year. I want to thank a couple of people. I want to thank Kathleen O'Connor. I want to thank Terry Keys for helping put on this great event. And when you think about it, in terms for cancer centers to thrive and survive and to achieve our dreams of really making an impact on cancer, not only in our state, but really nationwide, you need a bunch of things. And Lord knows I need a bunch of help. So we all need a bunch of help. And we need institutional commitments, and we're very fortunate, very blessed to have Dr. Mike Karff. Mike gave a couple of comments at the very beginning. I think he's had to go now, but you talk about institutional support. He's really been behind us all the way. He's the reason why I'm here. And with his vision for UK and for the Cancer Center, it's been fantastic. I'd like to thank Lisa Cassis, our Vice President for Research, who also gave some comments. Bob DiPaolo, our Dean of College Medicine, wonderful working with him. He was Cancer Center Director at Rutgers and now is our Dean, and unfortunately had a family event and couldn't be here either. In addition to strong institutional commitment to achieve our dreams, we need strong philanthropic support as well. And again, I feel very blessed and very fortunate to have fantastic Markey Cancer Foundation. You've heard from Sally Humphreys, who's the chair of the board, and Mr. Mike Delzadi, we recently recruited from MD Anderson. He is the president and CEO of the Markey Cancer Foundation. And I'd like to personally thank you all. Many of our board members are here in the audience as well, so thank you all for coming. And thank you for what you do on a daily basis. Okay. So I think what we're going to, this is a filler, I understand that. This is waiting until we get to the keynote address of Dr. Hunter. So I'll try to be fairly quick here. But what I want to say is it's a great honor, it's a great privilege to be able to stand up and talk about the successes of the Markey Cancer Center. And I think with one word sort of sums up the key or the key message to this whole talk, and that's growth, okay? What you'll see is growth in our clinical arena, growth in our research arena, and I tell people when they come and visit that our number one strength is our growth, but our number one weakness is our growth, okay? Because every day it's a bit of a challenge because it's putting in more and more patients into the same footprint. And, you know, luckily our infrastructure is increasing, we're getting more buildings going forward, which is great. But again, it's been remarkable to see the amount of growth that's happened. So just to recap a bit, we achieved NCI designation, as you all know. In July 2013, we put in our application in 2012, and we're fortunate first time around to get that. We became the 68th cancer center in the country to achieve NCI designation. There's been one since, so there's 69 total. Our dream, our goal, though, is to be a comprehensive cancer center. And with our application this time, and we'll see what happens. I mean, I don't think any other cancer center has been able to go from NCI designation to the first renewal going for comprehensive status, but we're going to try, and I think we got a great story to tell. So our mission, and I try to show this every chance I get because it truly is our mission, it's the reason why we come to work, it's the reason why we do our research, is to reduce cancer mortality in our state and region through a comprehensive program of cancer research, treatment, education, and community engagement with a particular focus on our underserved population of Appalachian Kentucky. So if you look at the Markey Cancer Center, we're very fortunate to be on a land-grant institution with all 16 colleges on a central footprint, okay? So that allows for a lot of great collaborations that a lot of institutions don't have, and we take great advantage of that. We have 112 total members. Our members come from 9 of 16 different colleges. We have 27 departments that are represented. All of our research programs have members from 2 or more colleges and at least 4 departments. Now, you all have heard me talk a lot about Kentucky and the problems that we have. Those of you who have not, I think it's at least worth us going over this again very briefly. But I think this really highlights the challenges that we face as a cancer center and as an institution in terms of our population that we deal with. And so the NCI is all about catchment area now. They want you to define the population that you take care of. And I really think that we've got this in spades. We're going to do great on this. So Kentucky is our catchment area. In Kentucky, 120 counties, 4.4 million people, about 42% are rural, 16.5% without high school diploma, and about 18.5% that live below the poverty level. But if you look at the population that we see, over 50% of our patient population comes from eastern Kentucky or Appalachia. So about half the state in terms of land mass, about 54 counties, less people, 1.1 million people, but highly rural. So about 82% of the population are rural. Almost 25% of this population are without high school diploma, and over 25% live below the poverty level. So these are some of the challenges that we face, is how to deal with a rural population that has literacy issues, poverty issues, and high cancer rates. And this slide illustrates this point as well. So unfortunately, we like to talk about how our basketball teams are number one. We don't talk as much about this problem here of our cancer, and I guarantee you if we talk more about it, more people will be doing things about it. So Kentucky, number one in the nation in incidence rates of all cancer sites. We're number one in the nation in cancer mortality. But as bad as and grim as these statistics are, it's even worse in Appalachian Kentucky that has some of the highest rates of cancer incidence and mortality in the country. So again, these are some of the challenges that we face, and this is why the Markey Cancer Center is so important to the NCI and to the nation, because of the niche that we serve, this underserved population. So I'm going to talk to you a little bit about, I'm going to start first with our clinical highlights and our growth. So if you look, I was fortunate to come in 2009. So in 2009, there were a little over 2,300 new cancer cases that came to the Markey, and this past year we will see 3,800 new cancer cases. That's an increase of 60% over the past 7 years or so. And if you look at our growth in our outpatient cancer visits, we've gone from a little over 62,000 patient visits to last year at 95,000, and this year we're projected to go over 100,000 patient visits. So a remarkable amount of growth, and this is what I mean by really trying to figure out how you're fitting in more and more patients into the same footprint. Some of the things that we've had to do, which again I feel very fortunate to have had a part in, we have had to seriously increase our support services. So when I first came in 2009, we had one poor social worker that was trying to take care of all the patients. And unfortunately, that's impossible. So we've greatly increased the size of our social work. We now have 6 dedicated social workers. They contacted over 7,000 patients in 1 year from July 2015 to June 2016. We've increased genetic counseling. Now, we will now have 2 genetic counselors. And the great thing about this is that they're not only serving Markey patients, but they're serving our affiliates as well through teleconferencing. We have two dietitians now as well. So we've really grown the support services, which we desperately needed to. The other thing, I mentioned infrastructure. So we're very pleased that our new Markey Cancer Center inpatient floor will be opening in December 2017. This is on track to open. And what that provides will be 63 more new inpatient beds, which is already too few. We probably see we probably have at any one time about 100 patients in the hospital. So we're going to have to figure out ways to further cohort our inpatients, and probably some of the Roach building will have to remain some inpatient space. But what this will be, 63 new inpatient beds, 32 of these will be for bone marrow transplant and for hematology. The other thing that we've expanded greatly is our integrative medicine services. This is under Dr. Connie Jennings' direction. And, 2 years ago, we had Jin-Jin-Jitsu, which was very popular with our patient population. But now we have a whole repertoire of integrative medicine aids to help our patients, which is very popular. I mean, this provides relaxation techniques for patients undergoing chemotherapy, et cetera. And also, this area is growing for us, so we're renovating space in the 3rd floor of Whitney Henderson that will house our new integrative medicine services. Patient satisfaction continues to grow up. Go up. I tell everybody I'm so fortunate and blessed to have a fantastic staff of nurses, of physicians. Everywhere you go across the town, people will talk about the great care that loved ones had at Markey Cancer Center. And what you see here is some dedicated work by Mark Filburn and others in the clinics to really further enhance our patient satisfaction. And this is in the face of, again, trying to cram in more patients into space. So that oftentimes a dissatisfier to patients. But the fact that we've been able to handle that flow and still have great satisfaction rates in our clinics has been a true testament to a lot of hard work folks have done. Some other highlights, we've established survivorship clinics for each of our clinical specialties. One of the great things about cancer treatments over the years is that more people are surviving, thank goodness. And so we have to have specialized services in order to take care of those survivors. We've introduced Emergent Patient Clinic, which is staffed by advanced practice providers, 2 clinic days per week. We've really run out of time, space for doing chemotherapy during the week, so we've really had to expand over the weekends now. And we've further expanded, so we've had 30% growth in our weekend volume over the prior year. And we're starting a new program with the College of, with pharmacy to establish home chemotherapy infusion options for cancer patients, which is increase further the patient satisfaction and decrease hospital days, because everybody would like to have their chemotherapy if they could at home. Okay, some of the research highlights and growth, because again, we've grown from a research perspective as well, which is quite gratifying. So if you look at this is our cancer-related funding. So in 2012, when right before our site visit, we had a little over $28 million in cancer-related funding, with about a third of that, with a little over a third of that coming from the NCI. Despite flat NIH budgets and flat funding rates, we've increased our funding level by 61%. So currently we have a little over $46 million in cancer-related funding, with a little over a third of that from the NCI. We have 4 outstanding research programs. We have Cancer Prevention and Control. This is led by Dr. Tom Tucker and Mark Dignan. And recently we recruited Dr. Tianyan Gao from the University of Indiana. Tianyan is a cancer genetic epidemiologist. She's going to be co-leading this Cancer Prevention and Control program. Dr. Tucker will then be focusing on this AD role. Drs. Jill Kalesser and John Thorson, we recently recruited Jill Kalesser from the University of Wisconsin. They're co-leaders of our DT program. We have a genomic instability and epigenetics program. It's led by Drs. John DeRazio and Peter Zhao. And Vivek Ragnikar and Rena Plattner lead our cancer cell biology and signaling program. We have 7 either existing or developing shared resource facilities at the Markey Cancer Center, and the efforts are being led by Dr. Heidi Weiss, who's our Associate Director for Shared Resources. And when you look at what we're doing in our catchment population, this is just a snippet. So this is not every research project that's ongoing in the Appalachian community. But what you see is a real dedication of the Markey Cancer Center to this underserved population. And in that regard, we've been very successful with CCSG supplement awards, and Dr. Vanderpool has already mentioned this in terms of some of her work with HPV vaccination. But what these are, so there are a number of benefits for being NCI designated, okay, many benefits. Increased growth, increased research support. But one great benefit is that you have access to these supplement awards that are only given out through a competitive process to NCI designated cancer centers. If you're not an NCI-designated cancer center, you can't get a supplement. And I think we've been very successful in getting a number of supplements to help support our research. A lot of this is going towards serving our underserved population. You see the Hpv Vaccination supplement, a national outreach network. We're the leaders in Region 1 of the Geographic Management of Cancer Health Disparities program. We have a population health assessments supplement, and recently have a cure supplement that is helping to support undergraduate students from UK who are from Appalachia, trying to help engender some enthusiasm regarding cancer research or cancer clinical care. So all told, total funding from our supplements alone since 2013 have been over $10 million. I'd like to highlight the fact as well that we recently received a large grant. It's the Center for Cancer and Metabolism. It's a COBRA grant, $11.2 million total. The multi-PIs are Drs. Dorette Sinclair and Peter Zhao. And basically what this helps to do, it helps to support cores, but it also helps to mentor and educate our junior faculty and to bring them up and to make them more competitive for R01 applications. So we currently have 4 junior faculty shown here who are part of the COBRA grant, which just started. Another aspect that I'm very pleased and very proud of is Dr. Kalesser is leading our efforts for an early phase clinical trial center. This has been a goal of ours as well for several years, and we now have dedicated space in the hospital that's being renovated. Hopefully this will be started in September 2017. And what this will do, it's a standalone clinical research center. It's got dedicated staff. It requires dedicated expertise in early drug development and in the delivery of these drugs. So it becomes a conduit for Markey Cancer Center translational science and support for our investigators who are interested in the delivery of novel therapeutics. So basically it allows us to do early phase clinical trials, some of the investigational drugs, again, that will only be available at NCI designated cancer centers. Some of those trials will be done in this new center. Also, I just want to highlight the fact our phase 1 study, phase 2 partnership with Ohio State. Dr. Suzanne Arnold received a supplement for phase 2 studies with Ohio State. Dr. DiPaolo, our dean, while at Rutgers, had what's called a UM1 grant that was for phase 1 studies. So he brought that with him, and we were able to merge with Ohio State. Now we're multi-PIs on this grant, and other participants include Cleveland Clinic and Case Western. What this has done is brought new trials to Markey Cancer Center. So it's made us a part of the phase 1, phase 2, the CTEP trials, part of the NCI. So again, we get new investigational compounds that nobody else gets except for NCI designated cancer centers. Again, I'm very proud of this. Our molecular tumor board, co-directed by Drs. Leicester and Rachel Miller in GYN Oncology. This was launched in October 2016 in Markey and is now statewide, such that physicians from across the state can call in if they have a difficult case and ask for advice. Basically, this utilizes a multidisciplinary team. The molecular tumor board analyzes molecular characteristics of cancers on a case-by-case basis. It then provides individualized recommendations for this treatment. And in March of this year, we held the first symposium on strategies for implementing precision medicine in clinical practice. And this was about 200 providers from across the state that came to this conference. I want to talk very briefly about our community outreach and engagement. Again, very proud of these efforts. If you look at what has happened with our affiliate network, which has grown significantly over the past couple of years, when we went in for our application in 2012, we had 7 affiliate hospitals, predominantly in central and eastern Kentucky. And through the great work of many people, including Dr. Tim Mullet, who's directing this program, we now have a total of 16 affiliate network sites, and they span both central eastern Kentucky as well as southern western Kentucky, so really throughout the state. I anticipate another 2, 3, 4 coming on within this year as well. And what this does, it really allows us to raise the bar of cancer care and prevention and screening throughout Kentucky. So it educates and it trains healthcare providers and staff as to the best practices and standard cancer protocols. So the chemotherapy nurses in Hardin, for example, are using the same protocols as Markey Cancer Center. We've rolled out the molecular tumor board to our affiliates as well as genetic counseling. We have joint conferences and symposia. We do community outreach and education, joint cancer screening events, and we also, Dr. Mullet and his team assist with them coming up to the standards of the American College of Surgeons Commission on Cancer Accreditation, which is, again, really raising the bar for cancer care throughout the state. In our research network, what we found is we were trying to do research-related projects with our affiliates, and that just didn't work because we were trying to cram in too much into one thing, basically. So we split this off. Our affiliate network is specifically for prevention, it's for education, it's for clinical care, but we split off now a research network, which we work with our partners to promulgate Markey research into these network sites. So we started out in January 2015. We had 4 research affiliates in eastern Kentucky. And we've since added 3 more in middle and western Kentucky. And I'm also proud to say that we're branching out into West Virginia because I think that's our duty as Markey Cancer Center to go into West Virginia as well. They have many of the same problems that we do in terms of high cancer rates. They don't have an NCI designated cancer center. So we're working on putting affiliate sites from Markey Cancer Center in West Virginia, and we have now 1 research network site in West Virginia as well. So we currently have 13 trials open, and what you see is it really spans quite a few counties. It spans 70 Kentucky counties, 7 West Virginia counties, and 1 Ohio county. And again, this has been efforts by Chris Dameron, who's here in the audience, and Tim Mullett. We have a community outreach program that's headed by Melissa Hounshell and Carla Repass, and this has been very instrumental as well in terms of going out and reaching a population. This program has reached over 11,000 people to date. Youth-focused educations as well as screening efforts have been the major focus of this. And finally, I want to talk about some other achievements for the Markey Cancer Center, but this is being wrapped up here pretty quickly. So marketing continues to be a big thing that we do, and thanks to Erin McElwain and her crew, has really served to get the great word about Markey out to a larger audience. I don't understand social media, but I don't do Twitter or anything else, but this has been something that Erin has really helped to promulgate for the center as well in terms of our social media presence. Other things that have happened over the last year, we had a moonshot event when other centers were doing that across the country as well, and I think we had a shout out from Joe Biden during his event. We had over 100 attendees. This was in June of last year. Erin and her team developed a new logo, consistent brand standards, redesigned our website. We had a Markey Can campaign that was launched in December. Our precision medicine conference was attended by over 190 attendees from Kentucky. Recently, Erin and Mike Delzati hosted the National Conference for Cancer Philanthropy and Communications Professionals from all across the country, and we had 435 participants, and that's the most ever for this meeting. It's a national meeting. Very proud that Markey was able to host that. And finally, if you've seen some of these very nice ads that started, I guess, with Kentucky Derby Day, launched a Markey TV campaign spot highlighting our precision medicine initiatives. It's called In This Room, started in May. Public relations, Allison Perry has been key in this regard. Again, getting the good word about Markey out to a larger group. We were fortunate that our Moonshot Summit event actually was covered by USA Today. Allison and her crew have also helped to broadcast our molecular tumor board and many other events, our COBRA announcement. And we were fortunate as well to host Dr. Lowy, the NCI director, in February of this year. And we got the good word out about that throughout the state as well. One of the probably only best thing that I did when I came here was to start an infrastructure called a Research Communications Office. And what this does, it provides support for Markey investigators. And really, it's branched out. A lot of investigators throughout the institution have utilized these services, but it's a very professional group of individuals who help with a number of different projects. I mean, about 25% of their projects are for grants. So they'll help put together grants. I mean, you can bring in, like I do, I've got a legal pad and I just write out grants, or you can have it in much better shape. But they will put it together and look professional, and it will help you get your grant. And not only grants, but they've helped a lot with our presentations, posters of Markey Cancer Center investigators. It's really become a fantastic infrastructure for our investigators. They provide a lot of support on the newly funded COBRA grant, and even helped the year previously on the CTSA renewal. 9 of UK's 16 colleges have used these services. 38 UK departments have used these services. And finally, I want to end with some upcoming events that we're getting ready for. Dr. Mullett and his crew are entertaining a visit from the Commission on Cancer. This will be June 8th. It's their triennial survey. We're looking to be a top performer, as always. So appreciate Dr. Mullett and his crew for this. Expression of Courage is in its 3rd year. It's really the brainchild of Ms. Carla Repass. And if you've never been to one of these events, it's quite an emotionally moving event. I would encourage you to come. It gets bigger every year. It really honors our survivors. It has art displays as well. And finally, so the looming thing that keeps me up at night is our Ccsd renewal. It doesn't seem possible that you know, 5 years ago, standing up here and talking about going for this and the angst associated with that, well, I can guarantee you there's just as much angst with this renewal application as there was with the first one. So our application goes in September 25, and then our site visit will be in early 2018. So finally, I'm just going to say, again, a note of thanks to everybody. This is a team effort, and really fortunate to be part of that team and to really go forward with these efforts. I truly think that we working together are making a difference in Kentucky, okay? Thank you very much. So speaking of the great efforts of philanthropy and how much that has been a great help for us. Ms. Lois Reynolds is going to talk about a new initiative that just started. In fact, we had an announcement yesterday, and you're going to make an announcement today. So again, we very much appreciate our board and everything that they've done for us in the Cancer Center.
Thank you. Markey Women Strong is a philanthropy program directed by women in the support of female researchers here at Markey Cancer Center and at UK Healthcare. Each Markey Women Strong member, and here's a representative group of our Markey Women Strong. This is our first year, and we're really excited that we have some of our members here as well. And if they come join me at this time, that'd be great. Each Markey Women Strong member donated $1,000 to join the 2017 Selection Committee. The Selection Committee reviewed 5 grant applications this year, and competition was tough, but we selected 2 outstanding researchers. Along with this award comes a $50,000 grant to support each recipient's work. It's my great pleasure and honor to introduce to you the recipients of the inaugural Markey Women Strong Distinguished Researchers Award. Dr. Kathleen O'Connor. Dr. O'Connor is a full professor in the Department of Molecular and Cellular Biochemistry and an Associate Director of Cancer Education at UK Markey Cancer Center. The O'Connor Laboratory studies breast cancer and the traits that make them more metastatic and less sensitive to current chemotherapies. Triple-negative breast cancer remains the most deadly form of breast cancer due to its aggressive nature, and even more worrisome, no target therapies currently exist for its treatment. Until these targeted therapies can eventually be developed, Kathleen's research focuses on exploring innovative and novel ways to use existing chemotherapies as an effective agent in fighting this breast cancer. Congratulations. Dr. Rena Plattner. A full professor in the Department of Pharmacology and Nutritional Sciences. The Plattner Laboratory studies melanoma, a highly aggressive and deadly form of skin cancer. Unlike most cancers, the incidence of melanoma is increasing, particularly in young women, and the disease remains incurable for many. Rena's work is highly significant because it will likely lead to new therapies for treating patients harboring melanomas that are resistant to current therapies and likely increase patient survival. Please join me in congratulating our inaugural Markey Women Strong Distinguished Researchers. Thank you.
So it is my extreme pleasure to announce the Leicester Memorial Lecturer. This seminar was originally the brainchild of the Leicester family, who wished to immortalize their memory of their wonderful wife and mother, Suzanne B. Leicester. This endowed lectureship was established by the family and friends of the late Susan B. Leicester, and they make this event possible. Mrs. Leicester left an indelible mark on the world. Serving as a clinical dietitian for those with long-term needs, she provided education to the Eastern State Hospital as well as to the nursing home patients in Eastern and Central Kentucky. Susan really loved educating her clients about the role of proper nutrition in preventing or treating conditions such as heart disease and diabetes, and believed strongly in its value, notes her husband, Dr. Thomas W. Leicester. This symposium benchmarks recent advances in cancer research and thus honors Susan B. Leicester by underscoring both her generous life and the brave battle that she fought against this dreadful disease. This year, we have the great honor of having Dr. Tony Hunter as our keynote speaker. Dr. Hunter started his training with his bachelor's at the University of Cambridge and went on to get his PhD there. He then moved to the Salk Institute and began his research as a postdoctoral fellow. And then when he was done, he eventually came back as an assistant professor. One of my favorite stories in science comes from Dr. Hunter's early work, and it's a tale of why scientists need to remain vigilant and observant. So when he first started off his lab at the Salk Institute, he was studying the phosphorylation of proteins. And in order to do this, you had to use large amounts of radioactive ATP. You phosphorylated your protein. You broke it down into the individual amino acids. Then you ran it out on this very thin layer of chromatography material that was up against a glass plate. And you stick it in a big buffer and allow that to be exposed. Then you take that and expose it to film. Well, when Dr. Hunter was working on the role of viruses in the Salk oncogene, he realized that after a long period of time, he kept reusing and reusing his buffer, as many of us do. With a lot of things to do, sometimes buffers just kind of get reused until things completely fail. And so Dr. Hunter actually did this, and one day when he went to develop the film, instead of seeing 2 spots, which represented serine and threonine, the 2 amino acids we need to be phosphorylated, a 3rd spot popped up. Well, a lot of us might have went, dang, I should have really replaced that buffer a long time ago. He actually realized that there was a 3rd amino acid that had an OH group that could be a receptor of the phosphate. He then had to now re-age his buffer and to find out exactly what the properties were that allowed him to see that 3rd spot, where he was able to identify phosphotyrosine. Back then, you couldn't just order phosphorylated tyrosine from Sigma, so he had to synthesize it and was able to use that as a reference spot to discover phosphotyrosine. Many of the drugs that we use now in the clinics and that are being developed are based on the fact that tyrosine kinases exist. And so with that ado, I'd like to have Dr. Hunter come up and give his talk.
Okay, it's my great pleasure to give the 2017 Susan B. Leicester Memorial Lecture. I last visited Lexington in 1986 and spoke about our work on tyrosine phosphorylation, and here we are 31 years later, and I'm still talking about tyrosine phosphorylation. So I got to start by just giving you some sort of brief general background on protein phosphorylation, which will be the topic of my talk. So as many of you know, protein phosphorylation is a reversible process in which a phosphate is linked to a protein covalently, often in an unstructured region between domains, by the agency of a protein kinase using ATP. And we would call this in the common vernacular a writer. That phosphate can be removed by a second enzyme, as you heard Tianyan talk about, a protein phosphatase that we can call an eraser. So this is a reversible process that leads to the phosphorylation of proteins, and that switches them into a new state. Phosphorylation can be recognized in many ways, but one way is through phosphobinding proteins, for instance, a protein with an SH2 domain, and these are known as readers. The major types of phosphorylation, although I'll be talking about a different type later, are the serine, threonine, and tyrosine phosphorylations, which create phosphate ester linkages like this. Now, phosphorylation is the most common type of post-translational modification, but you should realize that there are over 400 different types of post-translational modification known, with new ones still being discovered through the use of high-sensitivity mass spec. Now, phosphorylation is an extremely common event. HeLa cells have as many as 40,000 different phosphorylation sites in their 10,000 proteins, which means that on average, every protein has 4 different phosphorylation sites. And this is one of the challenges in the field. We really don't understand what all of these phosphorylations do. But one strong idea, or prevalent idea, is that post-translational modifications like phosphorylation is a mechanism to increase the complexity of the proteome. A typical vertebrate will have 20,000 genes, let's say. Perhaps these can make, through alternative splicing, 100,000 different proteins. It's a large number, but actually, given the complexity of the body plan and the functions of a mammal, 100,000 perhaps isn't really very many. But if you now include all of the different post-translationally modified states of these proteins, you can get 1 million or several million different proteins with perhaps subtly different functions. Just to give you some idea about other modifications, this database, Phosphocyte Plus, lists not only phosphorylations, it currently has over 275,000 different serine, threonine, and tyrosine phosphocytes. But it also has large numbers of ubiquitination sites, acetylation sites, with a total of closing in on 500,000 different post-translational modifications sites. And just to reiterate what Tianyan said, you can see here that serine phosphorylation is the most common, followed by threonine phosphorylation, followed by tyrosine phosphorylation sites. Now, as I already suggested, one major function of post-translational modifications is to serve as docking sites for a second protein that contains a PTM-specific docking domain. So, for instance, phosphotyrosine residues are recognized by proteins with SH2 domains. And I should point out that it's the 30th anniversary of the SH2 domain, which was first identified by the late Tony Pawson. But phosphoserine and phosphothreonine sites can also be recognized by specific domains, as can ubiquitinated lysines, sumoylated lysines, methylated and acetylated lysines. So this is a very common mechanism of inducible protein-protein interaction that is extremely important in signal transmission and gene regulation. Now, phosphorylation was the first protein modification to be discovered. I normally date this as 1906 from the work of Phoebus Levine, showing that vitellin, this egg protein, contains covalently attached phosphate, but some people prefer this 1883 paper showing that casein contains phosphate. The identification of the type of modification waited until 1932, the work of Fritz Lippmann, who synthesized phosphoserine, phosphothreonine, and phosphotyrosine to identify the linkage. First protein kinase activity was reported by Eugene Kennedy in 1954, and then it was really the seminal work of Ed Krebs and Ed Fisher, who showed that phosphorylation of phosphorylase leads to a change in its enzymatic activity, establishing phosphorylation as a regulatory mechanism. That was followed by Krebs's work identifying the cyclic AMP-dependent protein kinase, which demonstrated that second messengers can activate phosphorylation. Tyrosine phosphorylation identified in 1979, and all this ultimately has led to the approval of now a significant number of drugs that target protein kinases for treatment of human disease. So it was fairly early on that we realized that there would be a large family of the protein kinases that modify proteins. And this began to emerge in the early 1980s when the first sequences of protein kinases were obtained. I know you can't read this from the back, but this one here is the cyclic AMP-dependent protein kinase, a region of that catalytic domain. And this was actually sequenced the old-fashioned way. In other words, they purified the protein, cut it up with proteases, and stitched the pieces together again. But all of the others were sequenced by cloning. And a key observation was that made by Barker and Dayhoff, who showed that the sequence of the viral Salk tyrosine kinase was related to that of pKa. And at this point, I started to make alignments of these sequences, and it became obvious that the tyrosine kinases and the serine-threonine kinases all contain conserved motifs, which you can see here in these yellow and green bands that run up and down. And there was even one motif here, the HRDL motif, which distinguished tyrosine kinases from serine-threonine kinases. So this was done manually. BB stands for before blast. And actually, the human eye is pretty good at doing this. This alignment actually was published in the Scientific American. It's not really a journal you would expect to go to find a new discovery, but it was published in an article I wrote in 1984, and then it was published more officially in the Annual Review of Biochemistry. And subsequent work with Steve Hanks, who was at the Salk Institute at the time, analyzing over 100 protein kinase catalytic domains, we were able to identify actually 12 conserved motifs in the catalytic domain, which is usually around 300 residues, although can have larger inserts. And these domains can either be basically the whole protein, like for pKa or CDC2, or it can be part of a much larger protein here. And when the first structure of a protein kinase catalytic domain was reported by Susan Taylor's group at UCSD, again, this is pKa, it became clear that most of these motifs lined the catalytic cleft, where you can see a bound ATP molecule. Here's the adenine ring in here. You can see a bound ATP molecule with the phosphate ready to be transferred onto a substrate. And in fact, mutation of most of these motifs, single point mutations, kills the kinase activity of almost any protein kinase in this family. And these are commonly used as kinase dead controls. So through sequence gazing, it was becoming apparent about 30 years ago, shortly after I visited here, that there would be a very large number of protein kinase genes. And you can see how the numbers increase very slowly at first, and then suddenly accelerated in the late 1970s and early 1980s when cloning became possible. And at this point, I made the rather rash prediction that there might be as many as 1,001 protein kinases. It turns out that the human kinome, as we call its collection of protein kinase genes, has only around 550 protein kinase genes. But there are organisms which have significantly more than that. The eucalyptus tree, which is very common in Southern California, has 2,500 protein kinase genes. And the champion to date is paramecium, that little flagellated unicellular organism you may have seen swimming around under a microscope in high school, has nearly 3,000 protein kinase genes. So at that time I was talking about this, and I was at a meeting where one of the speakers was drawing cartoons, and this was the cartoon he drew of me with kinases trickling out of my beard, which has got significantly grayer since then. The actual kinome tree looks a lot more beautiful, and here is the tree that we generated in 2002, mostly the work of Gerard Manning, who was then at Sugen, a company in the Bay Area. And his analysis showed that there were 7 major families of eukaryotic protein kinases. Tyrosine kinase is appropriately on top, and all of these are serine-threonine kinases. This is the branch with the cyclic AMP-dependent protein kinase. These are basophilic kinases. These are the calcium calmodulin regulated kinases. And these are the CDKs, MAP kinases, which are proline-directed kinases. But in addition to these that all can be mapped into a relatedness tree, there are also small families of atypical protein kinases, which are either very distantly related in sequence or unrelated. And particularly important here is this PI kinase-related kinases family, which includes ATM, ATR, mTOR, you've heard a bit about, and DNA PK. These are all highly conserved protein kinases, huge protein kinases, several thousand amino acids in length that play key roles in either DNA damage response or nutrient sensing. Now, at that point, we had a total of 518 protein kinases. Interestingly, about 10% of them are missing key catalytic residues in those motifs I was pointing out. And these are being called pseudokinases. They seem to, most of them lack catalytic activity, but they can play extremely important functions. They are conserved in many cases. And it's believed that they function as scaffolds and assemble other proteins around them, sometimes other kinases. So there has been some progress since then. That was Kinome 1.0, was 2002. Gerard Manning has been keeping it up to date, and as of last year, he now has a list of around 550 protein kinases. All of the new protein kinases are remote or atypical. They're very distantly related. There are a couple of very interesting new protein kinases. FAM20C, a member of a small family of proteins, is the real casein kinase. This is the enzyme that phosphorylates secreted proteins like casein. It exists in the lumen of the Golgi, where it phosphorylates proteins that enter the secretory pathway. There's also a secreted tyrosine kinase, and then this family of histidine kinases that I will talk about. There were no new canonical tyrosine kinases. We'd found them all. But a couple of interesting proteins have been reported to be able to phosphorylate tyrosine, including pyruvate kinase M2, a metabolic kinase, which apparently can also phosphorylate tyrosine in some target proteins. So back to the tyrosine kinases, there are 90 human tyrosine kinases. 5 or 6 of them are pseudokinases. They fall into several subfamilies. Salk family is here, I guess, and then many of these are receptor tyrosine kinase families. And over half of them are implicated in cancer. either through activating mutations, like the original viral Salk kinase has an activating mutation in it, or they're simply overexpressed, and that certainly happens in types of cancer where genes are amplified, leading to overexpression. So there are a number of open questions about phosphorylation. I already hinted at one. There are thousands and thousands of phosphorylation sites. Are they all functional? And if so, how do we find out? It's possible, and people have suggested, that some of them may be silent, that they happen, but they're not deleterious to the cell, and so they're not selected against. Obviously, one can mutate these sites and see if you can identify a phenotype. It's going to be challenging with 40,000 to do in a cell. One can also look to see which ones are conserved through evolution, and this may be a better way to go. Many sites are phosphorylated at extremely low stoichiometry, 1 in 100 molecules. Are those significant or not? They could be if they're an activating phosphorylation, perhaps not if it's an inhibitory phosphorylation. And then another key question is, do sites act combinatorially? Are two sites on a protein inducing a different functional state from either alone? And that's something we know very little about. And in fact, there is no method currently for identifying which sites are occupied on a single protein molecule. None of the mass spec approaches do this for you because you always have to chop up the protein into peptides first. We know very little about how fast phosphates turn over in cells. When you do a blot with a phosphospecific antibody, you see what was on the protein at the time. You don't know how long that phosphate had been there for. There's some evidence that some phosphorylations turn over several times a minute. We have no idea whether that's important or not. It obviously could be, because it could set up some sort of oscillation. And then obviously a key question is how many of the 550 protein kinases might make drug targets. We know that several tyrosine kinases can be drug targets. Gerard Manning's estimate is that about a third of all protein kinase genes are potentially involved in human disease based on, as I said, mutations or expression differences. And most of these are implicated in cancer, where we're continuing to find new but rather rare point mutations in the kinome in different types of cancer. And it's actually through sequencing analysis that these are being discovered. And the BRAF tyrosine kinase, serine threonine kinase, which is a driver in melanoma, was actually identified as a mutant kinase through genome or kinome sequencing efforts, and has subsequently been targeted with a very selective kinase inhibitor. So currently there are 36 kinase inhibitors. 26 of these are tyrosine kinase inhibitors approved as cancer drugs. And this, I'm afraid, is really too dark to see, I expect. But Doriana Fabro, who's at PCure Therapeutics, has been keeping a tally of the approval of kinase inhibitors as drugs. And you can see, starting, and I can't read this now, but somewhere 2001 was the approval. Here it is in a box. Imatinib, the BCR-ABL tyrosine kinase inhibitor known as Gleevec, that is very successful in treating particularly early-stage chronic myelogenous leukemia. But you can see there have been increasing numbers over the years, and there are many more, apparently up to 400 clinical trials going on with kinase inhibitors, and so there will be many more kinase inhibitor drugs to add to this armamentarium. So with that not so brief introduction, let me just tell you two short stories about work that's currently going on in the lab. The first is on pancreatic cancer, which, as you know, is one of the most deadly cancers. Sadly, it's almost always diagnosed too late. It's almost in the terminal stage that has metastasized, life expectancy of less than a year. And I got introduced to this as part of a Stand Up to Cancer pancreatic cancer dream team, which are teams of scientists, both clinicians and basic scientists, that are distributed around the country and now even in Europe as well, which bring people together to try and develop new therapies for particular targeted cancer, in this case pancreatic cancer. And our job at the Salk Institute, where the team was myself, Jeff Wall, and Ron Evans, was to try and understand the contribution of stromal cells, non-tumor cells, in pancreatic cancer. So let me just remind you then that pancreatic cancer has a fairly well-defined genetic progression where there's a mutation in KRAS, an activating mutation, further mutations in CDK2N, 2A, loss of function in P53, and often SMAD4. And the morphology goes through a series of characteristic changes where you form these so-called PANINs, pancreatic cancer in situ, which ultimately become disorganized when they reach the terminal stage and then become invasive, and they can infiltrate and move and ultimately perhaps disseminate into the bloodstream to generate metastasis at different sites, in this case, often in the liver. So I should remind you then, as I've implied, that solid tumors are a community of cells. They're not just tumor cells. They contain not only the tumor cells, but also blood vessels where there are endothelial cells and pericytes. They contain a number of immune cells and inflammatory cells, macrophages, T and B cells of various sorts. But also fibroblastic cells, mesenchymal cells, like stellate cells that I'll tell you about. And so these cells are all interacting. They're talking to one another, and they're presumably supporting one another. And pancreatic cancer has a very unusual stromal cell population. It's sometimes called a desmoplastic response, in which nests of tumor cells are surrounded by a very dense stroma that's generated by these stellate cells as they switch into an activated myofibroblast state. And this forms a barrier both to the vascular system but also to drug penetration, which is one of the reasons why it's a hard tumor to treat. Now, stellate cells are a resident population in the pancreas and liver and kidney, and they have a sort of defensive function. They respond to injury and to inflammation, and they have a characteristic shape, very star-like. And then, as I said, in response to injury or inflammation, they become activated and myofibroblasts-like and start to pump out various proteins, matrix proteins, but also cytokines. And so they become a focus for potential therapeutic intervention. So we set out to study the interactions between the stellate cells on the one hand and the tumor cells on the other hand, with the idea that the stellate cells would make factors that act on the tumor cells, and in turn the tumor cells would make factors that act on the stellate cells. And we wanted to try and understand what sort of paracrine factors were being secreted by these two cell types. And the idea of focusing on secreted factors was that they could potentially be targeted by protein drugs like monoclonal antibodies. So to do this, Yu Shi, a postdoc in the lab, set out to identify the secretome of both these cell types. So the secretome is the collection of secreted proteins. So he took a semi-immortalized human pancreatic stellate cell line and a couple of established human pancreatic cancer cell lines, MIA-PACA2 and PANC1. And he grew these cells in serum-free medium for 24 to 48 hours, and then carried out mass spec analysis on the proteins in the conditioned medium. So the conditioned medium was treated, and ultimately the isolated proteins were digested with trypsin and analyzed by mass spec by Ru Tian, who was a postdoc in Tony Paulson's lab at the time, but is now in Shenzhen in China. We were also interested in what signaling processes were activated when the conditioned medium of one cell type was used to treat the second cell type. And so that's what's illustrated here, where we used actually methyl labeling to do the mass spec analysis. And we focused on tyrosine phosphorylation events because they are often very characteristic of signaling pathways. And so we isolated the phosphotyrosine peptides from the proteins in the cells that following trypsin digestion and identified these sites. So these secretomes are complex. They both have about 1,000 proteins in them. This is typical of what you find when you analyze conditioned medium from cells. So they're fairly reproducible. And what we were interested in was proteins unique to each cell type. And so there were around 140 proteins unique to the stellate cell conditioned medium and about 400 unique to the cancer cell conditioned medium. And this was, as I say, the work of Yu Shi and Ru Tian. So when you do this, you get long lists of proteins, and you have to sort of focus on particular types of protein. And so for the stellate cells, in both cases, we were interested in potential growth factors, cytokines. For the stellate cells, you can see there are very large numbers of extracellular matrix proteins identified. This is what we expected. There are also a number of proteases and inhibitors that would be involved in remodeling the extracellular matrix. There were very few. There was only one extracellular matrix protein unique to the cancer cells. There were a number of interesting growth factors and cytokines produced by the stellate cells, and we focused on LIF, leukemia inhibitory factor, which is a member of the IL-6 family, as I will show you. But there were some other interesting proteins like CXCL12, which inhibits T-cell entry. The tumor cells, I won't say much more about the secreted proteins, but we have done some work on PDGF, which is produced by the pancreatic tumor cells and acts back on the stellate cells. So what signals in the tumor cells are activated by the stellate cell conditioned medium? So we looked at the phosphotyrosine signature, and the main event we saw when either of these two tumor cell lines were treated with stellate cell conditioned medium was STAT3. STAT3 tyrosine 705. And we know that this is an event that's stimulated by many cytokines through cytokine receptors. And others have reported roles for IL-6, another family member, and STAT in pancreatic cancer. Now, so the question is, we have this complex mixture, which protein, which receptor system, and which ligand were involved. And so for this purpose, Yu Shi set up I think this is a PANC1 cell, stably expressing FLAG-STAT3, stimulated with conditioned medium, pulled out FLAG-STAT3, and ran mass spec on it. And the proteins he got out were the most common proteins, and there were many, but it was the LIF receptor, GP130, which is a co-receptor for the LIF receptor, and JAK1, the tyrosine kinase, that phosphorylates STAT3. So this was the main reason we focused on LIF. So as I said, LIF is a member of the IL-6 family of cytokines. It's an interesting cytokine because it is important for the self-renewal of many types of stem cells. So this is the sort of function you might want in a tumor, I guess. In fact, any of you who work on ES cells in culture will be using LIF to keep these cells alive. These cytokines use heterodimer or heterotetrameric receptors in which there's a ligand binding subunit, in this case the LIF receptor for LIF, and then GP130, which is regarded as the signaling subunit. But each of them has a unique receptor subunit. And these receptors activate the JAK tyrosine kinases, which lead to phosphorylation of STATs, migration of STAT dimers into the nucleus where they can drive expression of many genes, including stem cell genes like SOX2 and OCT4, but they can also activate other pathways like ERK and PI3 kinase. A little bit is known about the potential role of LIF and the LIF receptor in cancer. Interestingly, LIF knockout mice are viable, although LIF receptor knockout mice are not. And there's some evidence that LIF can induce EMT, and also that it's upregulated by KRAS, suggesting something about pancreatic cancer. So our secretive analysis has sort of led us to the idea that there might be this reciprocal loop between the stellate cells producing LIF, activating the LIF receptor in the tumor cells, which then leads to the expression of PDGF, which acts on the PDGF receptor in the stellate cells. So you form this positive feedback loop, if you like, maintaining both the tumor cells in a proliferative survival state and activating the stellate cells. So if we look at a series of different human pancreatic cell lines, many of them are sensitive to LIF as monitored by STAT3 phosphorylation. So the ones in green all respond to LIF, but others like this red one respond better to IL-6 than to LIF. If we take stellate cell conditioned medium and neutralize either LIF or IL-6 with antibodies and then test that conditioned medium, you can see for this cell line, KP4, inhibition of LIF causes a large decrease in STAT3 phosphorylation, whereas inhibition of IL-6 doesn't do so much, but if you combine the two, you get an almost complete loss. So LIF and then IL-6 are the key factors in the stellate cells. Okay, so we have a target. Would interfering with LIF in a tumor situation have any consequence? So for this purpose, we've used the so-called KPC mouse. This is the mouse that was developed by Dave Tuveson when he was with Tyler Jacks, in which PDX1 is a driver for Cre expression. This is driven in the epithelial cells of the developing pancreas, and this leads to expression of an activated RAS and the loss of deletion of p53. And these mice with both alleles of p53 floxed develop pancreatic cancer and die by about day 60, a very aggressive, rapid disease, so potentially difficult to treat. So basically, we identified a neutralizing LIF monoclonal antibody, originally made by Genentech, it's D25, and tested it in this system. We also added a luciferase reporter. So this just shows you the characterization of the antibody. We have recombinant human LIF and recombinant mouse LIF, and here is the amount of antibody we added, and you can see that we can neutralize both of them with this antibody, but it takes actually a lot more to neutralize mouse LIF than to neutralize human LIF. Not surprising, this antibody was raised against human LIF. So we tested the effect of different doses of the antibody in the KPC model, and here is a mouse treated with 25 mg per kg of the D25 antibody, stained. The tumor is stained for STAT3, and you can see there are tumor nests where there is still strong nuclear staining for phospho-STAT3, but most of the tumor cells lack STAT3. So we used 25 mg per kg in this treatment regimen, where we've combined it with gemcitabine. It's a nucleoside analog that is the standard of care for pancreatic cancer. It's really not very effective at all, but we've used the anti-LIF antibody administered in these cycles together with gemcitabine at 50 mg per kg. And gratifyingly, we see a significant extension in survival of the mice that are treated with the anti-LIF antibody compared to a control IgG. It's about a 20% increase in survival, which is pretty good in this model system. We also see a significant decrease in tumor weight with the anti-LIF antibody. And we had asked 3 independent pathologists to do analysis on these tumors, and they agree that the tumors from animals treated with the anti-LIF antibody are significantly more well differentiated. So we think we're promoting differentiation of the tumor cells. Consistent with that, we see, and this is work done together with Tanish Therer at UCSD, we see a decrease in 4 different stem cell markers in the on cells isolated from the tumor in the animals treated with the anti-LIF antibody. So potentially then, the anti-LIF treatment is decreasing the number of stem-like cells in the tumor, and that's leading to increased differentiation. As a second way of checking whether LIF signaling is important, we've used a conditional knockout of the LIF receptor in the same system where it's going to be knocked out in the epithelial cells, the ductal cells of the pancreas. Now, we've actually shown that the LIF receptor, using RNA scope, this is 2-color in situ hybridization, is predominantly expressed in the tumor cells. You can see the red color here is the LIF receptor. It's in the tumor cells, and that contrasts to LIF, again using RNA scope, where it's predominantly expressed in the stroma with a few tumor cells expressing LIF. And interestingly, you can see that most of the cells that make LIF are very close to the tumor cells, consistent with paracrine signaling. So if we run this experiment, and you should look here at the green line, these are the mice in where both alleles of the LIF receptor were floxed. You can see, again, a significant extension in lifespan compared to the control or the mice with a single floxed allele of LIF receptor. So I think this is pretty consistent with LIF being derived from the stromal cells, stellate cells, acting on the tumor cells to promote their proliferation and survival. So obviously this is all very nice. This is in a mouse. We know that mice are not the same as humans, and there are many examples where you can cure cancer in a mouse and it doesn't work in humans. So the next thing we've done, and this is still in process, is to look to see whether we can find evidence for LIF involvement in human pancreatic cancer. And so we collaborated with Tim Donahue at UCLA to obtain pancreatic cancer tumor samples and carried out an ELISA analysis for LIF expression. And you can see here are the normal controls with very low levels of LIF. You can see here that, although there's quite a wide range, in general, there is a significant increase in LIF in the human pancreatic tumor tissue. And interestingly, also a hint of an increase in LIF in chronic pancreatitis tissue. And that's consistent with the fact that if we treat mouse with cerulane, which induces pancreatitis, we see a strong increase in LIF, both in the tumor and actually in the serum. And in that regard, we can see, although we don't have many controls here, we can see a significant level of LIF in the serum of the human pancreatic cancer patients. So we're hoping that LIF levels could be used potentially as a diagnostic, or certainly for assessing response to therapy. We've also developed, in collaboration with Ruijin, who's now in China, as I said, a targeted mass spec-based approach to look not only at LIF, where you can see very high levels of LIF protein in tumor tissue not seen in normals, but we can measure levels of any other protein of interest directly in tumor tissue using this targeted mass spec approach where one isolates glycoproteins first. So I think we're very excited by this, that we've identified leukemia inhibitory factor, or LIF, as a potential therapeutic target for human cancer, pancreatic cancer. We have reasonable evidence that the protein is upregulated in pancreatic cancer. And now what is needed is to develop a humanized anti-LIF antibody for therapeutic, for clinical trials. And there is a company, Northern Biologics, in Toronto that actually has such an antibody, and we're going to try and collaborate with them to see if we can use this in conjunction, anti-LIF antibody in conjunction with standard of care. So, how am I doing? Not too bad. So I'm going to switch gears completely to talk about something new and exciting going on in the lab that at the end you'll see also has a cancer connection. So, for those of you who don't remember, this is the structure of tyrosine. It has an aromatic ring with its phenolic hydroxyl group here, and phosphate goes onto this hydroxyl group to form phosphatyrosine. This is a phosphate ester. It's chemically very stable. Now, in addition to the 3 hydroxy amino acids, 6 other amino acids can be phosphorylated, and these include the 3 basic amino acids, histidine, lysine, and arginine. Histidine is interesting because it's another ring containing amino acid. It's an imidazole ring. It has 2 nitrogens, and each of those can be phosphorylated. You can form 1 phosphohistidine or 3 phosphohistidine. Now, these are phosphorus nitrogen bonds, and these phosphoramidates are chemically very unstable. They fall apart when you heat and at any temperature below, any pH below around 6. So while you may not have heard much about histidine phosphorylation, it's been around quite a long time. The first report of histidine phosphorylation was in a bacterial protein in 1962 by Poel Boyer's group. This was a phosphoenzyme intermediate. But it's well documented in bacteria where there are these so-called 2-component signaling systems that respond to external signals. There's a receptor protein that becomes autophosphorylated on a histidine, and this phosphate is then transferred onto an aspartate in the regulator protein, usually a transcription factor. And so this is actually a phosphohistidine phosphoaspartate relay. But there is phosphohistidine in eukaryotes. A number of enzymes have a phosphohistidine intermediate, like phosphoglycerate mutase, but also other proteins, which I'll show you in just a second. Now, other kinases, protein kinases that can phosphorylate histidine, well, a recent report suggests that these 2 proteins, NME1 and 2, also known as nucleoside diphosphate kinases, can phosphorylate histidine. And these are enzymes that normally utilize ATP to rephosphorylate nucleoside diphosphates to regenerate the triphosphate. These enzymes use a 1-PHIS enzyme intermediate to transfer phosphate to another nucleoside diphosphate and also to proteins. There's a family of 10 of these nucleoside diphosphate kinases, or NMEs as I'll call them, in 2 different groups. And we know these 2 can be protein kinases. Maybe some of these others can as well. There are at least 2, and I'll tell you briefly about a third, phosphohistidine phosphatases. So there are kinases and phosphatases. So in principle, this can act like any other phosphorylation as a reversible regulatory mechanism, with kinases as the writers and phosphatases as the erasers. Now, in bacteria, as I said, there are these 2-component systems which have a receptor-like histidine kinase that autophosphorylates when something binds to the extracellular region. And then when autophosphorylated, this transfers the phosphate to an aspartate in a response regulator to give you an output. Now, these systems do not exist in metazoans. Instead, they appear to have been replaced largely by tyrosine kinase signaling. Now, there are a few proteins where phosphorylation of histidine has been characterized as regulatory, particularly these 3 channels, 3 ion channels, where Ed Skolnick's group has shown that phosphorylation regulates ion channel activity, and particularly this histidine 358 in this calcium channel that's potassium channel that's activated by calcium. One particularly interesting phosphorylation is histone H4. It's phosphorylated at histidine 18 predominantly. This is conserved in all eukaryotes. It's in the tail of H4. It's got to be doing something important, but no one has any idea what it does. And this sort of summarizes most of what we currently know about histidine phosphorylation, and I know you can't read it, and I can't even read it, but if anyone's interested, we just published this in a review in Current Opinion in Cell Biology. But it illustrates that histidine phosphorylation events occur in all parts of the cell, from the nucleus to the membrane. Now, it's challenging to study histidine phosphorylation. As I told you, it's heat labile and it's acid labile, which means you can't use acid hydrolysis to identify it in proteins. And that's really been what's held back the field for all these years. 25 years ago, we set out to try and make antibodies specific for phosphohistidine and failed. And we believe we failed because the phosphohistidine antigen we used was simply too unstable in the animal. And so we knew what we needed then were stable analogs. And again, I'm sure you can't really see this, but Tom Muir's group and Mike Webb's group developed these phosphoryl triazolyl analogs in which, you can't really see this, the phosphate is linked to a carbon in an imidazole-like ring, mimicking the 1-phosphohistidine and 3-phosphohistidine. And we've used these as immunogens embedded in degenerate peptides that have random Aller and Gly residues on either side, a cysteine at the N-terminus to link this protein to KLH, this peptide to KLH. And then we immunize rabbits, and I should point out the chemistry was done by Jacques Marget's group at Sanofi. And this was the work of Steve Foos and Jill Meisenhelder in my lab. We immunized rabbits with either the 1-PTZA library or the 3-PTZA library and looked to see if we got antibodies. And much to our delight, we did get antibodies that recognize proteins we knew to contain phosphohistidine. So as a 1-PHIS containing protein, we chose NME1 itself. I told you it has a phosphoenzyme intermediate. And here you can see that the serum raised against 1-PHIS recognizes NME1, but not phosphoglycerate mutase, which I also told you has a phosphohistidine intermediate, but this is a 3-phosphohistidine, and you can see that the 3-phosphohistidine antibodies detect this, but not NME1. I told you that the phosphohistidine is unstable to heating, so if you boil the samples in sample buffer, almost all of the signal on PGAM goes away. So you might be asking me, well, how do we run gels if you can't boil the samples? Well, the answer is, probably not known to many of you, if you simply incubate your lysate with, or immunoprecipitate with sample buffer on ice, it equilibrates perfectly well with SDS. You don't need to boil your samples most of the time. And so you can run gels in the cold and do blots in the cold, and you can identify these labile proteins. So we went on and used the spleens from these rabbits to generate monoclonal antibodies. And we have a whole series of monoclonal antibodies that recognize 1-phosphohistidine or 1-PTZA, and another set that recognize 3-PTZA. And we use them here, for instance, one of the 1-PTZA monoclonals, SC11, to blot a series of pancreatic tumor cell lines, part of our pancreatic tumor project. In each case, there is a boiled sample and an unboiled sample, and you can see that in most cases in the boiled samples, most of the signal has gone. In the unboiled samples, you can see the major bands are NME1 and NME2 themselves, but there are a number of other proteins that might be of interest. There are 3 phosphorhistidine antibodies now used to blot either E. coli lysates or 293 cell lysates, show a lot of bands that go away with heating. And so we think probably 3-phosphohistidine is the major type of histidine phosphorylation in cells. And here we've used 2 different monoclonals, and we have 3 unique 1P His antibodies and 4 unique 3P His antibodies, and some of these are actually now available commercially if anyone's interested. We've done a number of things with these antibodies. For instance, we've used them for immunostaining, in this case HeLa cells, and the green so is the staining with the 3P His antibody, SC39-4. You can see there are sort of nuclear speckles. This might be the histone we're recognizing here, H4. You can see these 2 interesting dots here in these early prophase cells, and by anaphase you can see staining of the spindle poles. So we think histidine phosphorylation could be playing an important part in mitosis. We collaborated with Ed Skolnick actually to show that this channel that he's been working on, this potassium channel, KCA3.1, contains phosphohistidine. He deduced all of this indirectly. He identified this histidine 358 as the site of phosphorylation, but never shown directly that it was phosphorylated. This is phosphorylated actually by NME2, NDPKB, and it's dephosphorylated by the PHPT1 histidine phosphatase. And the phosphorylation which opens this channel is important in T cell activation because it allows more calcium into the cell as the potassium exits. So I'll just show you 1 or 2 pieces of data where he analyzed the potassium channel. In this case, it's a tagged potassium channel expressed stably in Jurkat cells, T cells. And he's also knocked down PGAM5, which is another phosphohistidine phosphatase. And you can see either in the controls or in the knockdown particularly, there's a strong 3P signal in the GFP channel IP, but no 1P His signal. Conversely, if you look at NDPK, there's a weak 1P His signal and a stronger 1P His signal in the PGAM knockdown cells. So we can conclude actually that this site is exclusively phosphorylated on the N3 position, but we don't know how this selectivity is achieved. How does this phosphate regulate activity? Well, this was again work that Ed Skolnick and Steve Hubbard did in collaboration with using our antibodies. And they've come up with evidence that this N3 phosphate prevents copper binding. I'm going to skip that. So this channel, the KCA3.1 channel, exists as a tetramer in the membrane, and the 4 C-terminal tails form a 4-helix bundle that coordinates a copper ion via this histidine. When calcium levels rise, the pre-bound calmodulin undergoes a conformational change which allows this histidine to be phosphorylated, and once it's phosphorylated, it can no longer bind this copper ion here. So the idea is the copper ion is inhibitory by holding the channel in a closed state. Once it's phosphorylated, this can now open up. So we think it might be a general principle that histidine phosphorylation regulates metal ion binding. The other thing we've done is to use our antibodies to try and identify the phosphohistidine proteome. So we've used our antibodies to affinity purify proteins from fully denatured lysates, cells lysed in 6 molar urea at pH 10 to stabilize phosphohistidine, and identify proteins that are selectively enriched by the antibodies. And we found about 800 proteins, so this could be quite a common event. They include many proteins involved in RNA biology, RNA processing, RNA splicing, ribosome biogenesis, translation. So that's potentially interesting. Yeah, and another set of proteins involved in the cell cycle, so consistent with our mitotic staining data. We did find histone H4, the NMEs, PGAM, ATP citrate liars were enriched as expected. But what we really need to do is to map the sites of histidine phosphorylation, because that's where we have to go to do functional analysis. And this is technically very difficult because under standard phosphoproteomic conditions, phosphate falls off. You use acid buffers for separating the peptides. The phosphate is also lost upon bombardment in the cell. But a postdoc in the lab, Kevin Adam, has actually developed better methods for enrichment of PHIS peptides and analyzing, and has now identified 196 phosphohistidine sites in 293 cell proteins. And so we're on the way to a phosphohistidine proteome. So there are a number of open questions. How big is the phosphohistidine proteome? Could be as many as 1,000 maybe. Under what conditions is histidine phosphorylation regulated? We don't really know that yet. Are there other histidine kinases in addition to the two NME family members? Are there other phosphatases? How can one study this process? One can mutate the histidine, but there isn't a good non-phosphorylatable analog. People have used asparagine. It's isosteric but not charged. We're collaborating with Lei Wang at UCSF to try and develop stable, unnatural amino acid analogs of phosphohistidine so we can incorporate them into proteins at specific sites through UAG suppression, and that's underway now. And then, obviously, the main question is how does histidine phosphorylation regulate activity? Are there phosphohistidine binding domains? We're looking for those, like phosphotyrosine binding domains. Does it work through local charge effects, or does it potentially regulate metal ion binding? Okay, so in the last couple of slides, I'll tell you about some very exciting work on the possible role for histidine phosphorylation in cancer, which has been made possible using our antibodies. This is collaboration with Mike Hall in the Biocentrum in Basel, work of his postdoc Sravant Thindupur. And what they've been interested in is hepatocellular carcinoma, and they have set up a mouse model in which there is liver-specific double knockout of TSC1, the subunit of the Reb gap, which regulates mTORC1, and PTEN, the lipid phosphatase that dephosphorylates PI3P. And these mice develop hepatocellular carcinoma by 16 to 20 weeks, you can see here. So they carried out a proteomic analysis on these tumors to see what proteins were changed, focusing on proteins involved in protein phosphorylation. And they came up with a list, but among the top hits were increased levels of NME1 and NME2, and decreased levels of LHPP. This stands for lysine histidine protein phosphatase. So this suggested that maybe there was going to be increased histidine phosphorylation in these hepatocellular carcinomas. Basically, this just shows you the downregulation of LHPP, upregulation of NME1 in tumors. So excitingly, they do see, well, first of all, let me show you that LHPP, recombinant LHPP, reduces the 3P His blot signal. Compare this box to this box when used to treat liver tumor cell extract. But what I was going to show you was that there is a significant increase in 3P His containing proteins in the tumor cells. Again, these are the Boyle controls, but no real change in 1P His except an increase in NME1 itself. He used an adenovirus to re-express or to overexpress LHPP in a cell line derived from one of these mouse hepatomas. And you can see here that there's a significant decrease in the level of 3P here. So if you re-increase the expression of this phosphatase, that reduces the level of histidine phosphorylation. If he now introduces LHPP expression using an adenovirus vector injected into the tail, he significantly decreases the number of tumors in these mice. So all this is suggesting that histidine phosphorylation is playing an important role. And then finally, he was able to show that if one takes tissue from human hepatocellular carcinomas, comparing normal liver with tumor tissue, you can see there's a significant increase in the level of 3P His in the human hepatocellular carcinoma tissue. So this suggests then that histidine phosphorylation is playing a positive role in hepatocellular carcinoma, and that LHPP, the phosphatase, is acting as a tumor suppressor. What they have found by analyzing patient RNA data is that patients with low LHPP levels have worse prognosis. And what we need to do now then obviously is identify the proteins that are getting phosphorylated on histidine to really establish that this is involved in hepatocellular carcinoma. I'm going to skip that and just thank the people who did all the work in the lab. Yushi drove this pancreatic cancer project. He came to the lab, and luckily he wanted to do this project. He's been working on it for 5 years. There's no paper yet, but soon we hope. It's being submitted, I think, next month, with help from Ruijin, Tim Donahue, Tanish Saraya, and funding from the NCI, Stand Up to Cancer, the Lustgarten Foundation, and the Helmsley Charitable Trust. The work on PHIS antibodies, spearheaded by Steve Foos with help from Jill, Aaron, Lee, and Kevin. We collaborated with Ed Skolnick's group and Mike Hall's group. I should say this work on phosphohistidine was not funded by a regular grant until recently, and not until we had the antibodies was anyone interested in funding it. It's often the way science goes, right? And we're now trying to make phospholysine antibodies. Thanks very much.
Question over here.
Oh, hi.
Histidine is known to be ubiquitinated. Are there any examples of interplay between ubiquitination and phosphorylation on histidine?
It's a good question. So you all heard that. Is it possible there's some crosstalk between histidine ubiquitination and histidine phosphorylation? We haven't looked. It's a good idea. I think we need to identify some histidine sites first, and then we can begin to look. It may be a question of stoichiometries. We think histidine phosphorylation is not a very high stoichiometry, so how well it would block ubiquitination and vice versa, I don't know, but it's certainly something worth looking at, yes. Yeah,
So
yeah.
are there any adapter proteins combined to phosphorylate histidine residues?
Yeah, so are there any proteins that selectively bind phosphohistidine? Yes, we've begun to look, so we've used our phosphohistidine peptide libraries to try and fish out proteins selectively. So far, we haven't found anything. We're playing around with some atypical SH2 domains to see if they might bind phosphohistidine. We have hints that they might. Whether that will be important, that's all in vitro, whether that will be important in cells, we don't know. But SH2 domains are attractive in the sense that we know they bind. It would be 3P His, which looks very like phosphotyrosine, so that's a potential, yeah. Okay, you all want your cocktails.
So each year when we have our Markey Cancer Research Day, we like to give you a few parting gifts to remember us by. This is a mint julep cup, so you can remember Kentucky. That's engraven with your name and Markey Cancer Research Day as your keynote speaker.
Thank you very much. Try and get it out of here without breaking it. There it is. It's very nice, yes.
And we've also paired with one of our local breweries that many of us like very much. Country Boy Brewing here in Lexington's a great story of a small group of businessmen that got together and decided to revolutionize microbrewery in Lexington, Kentucky. They are very supportive of the Cancer Center and cancer research as a whole, and they pair with the Kentucky Cancer Foundation. And they have a Country Boys and Girls Against Cancer event every year, and they've given us the glasses as gifts for our participants.
Thank you very much. I can use this at home. That's great.
Okay, thank you so very much.
Thank you. It's been my pleasure. I guess I should.
On stage, and we will get a picture and as well as give you your glasses. So we'll start off with the clinical sciences. Of course, we have 8 categories. We have clinical science and population science as one category, involving graduate students and postdocs. Our first place in the graduate student clinical sciences is Markey on the complex interplay between immune status and precision nanomaterials. Our second place was a poster authored by Aman Chowdhury and presented by Elizabeth Behrens. If I could have both of you to come up. And that's our second place. For our postdocs in clinical science, our first place poster was by Anthu Lee. The ACS NSQIP-based risk score predicts the early readmission after gastrectomy. And our second place is Nasser Jafari, and the activation of FASN SPHK axis promotes the metastatic potential of primary colorectal cancer cells. So all of you, if you could come on up. Our next category is the basic sciences. So our graduate student first place was one of our speakers last year. She had a posse around her poster today. It was very popular indeed. Katie McKenna. In the splenic microenvironment is important for the survival and growth of B-cell chronic lymphocytic leukemia in mice. So Katie. And our second place is Dustin Carroll. A novel redox-based therapy targets for malignant cellular redox state. Dustin. Our next category is the postdocs in basic science. Our first place is Rashimani Tripathi in the C-ABL-ARG modulation of cathepsin secretion via the SP1 NF-kappa B transcription factor activation. And our second place is Nikhil Hibar with a naturally generated PAR4 decoy overcomes therapy resistance in tumors. And a couple years ago, we had an overall winner. So the person who got the best score on their poster judging, and that is our own Peyton Stevens with the overall award. So if you'll come forward. None of this at all would ever be possible without the amazing talent and grace of Terry Keyes. You all know him as our education liaison. I've been working with him on this for 8 years. Our very first one, we had a minimum of 4 weeks to put everything together. We actually got together and thought of a venue, and we said we should do the Singletary. And I can't begin to tell you how many negative remarks we had about having to hoof it all the way over to Singletary. But in our defense, we were thinking pretty far in advance. We continued to grow this program until we now fill all of the Singletary. And so I'd like to have a very big round of applause for Terry and all the amazing work that he does in putting this together.
Thank you. I forgot it was my turn.
Okay, and last but not least, the Markey Cancer Center loves to acknowledge all of those of us that work hard every single day to make the difference in Markey. And so I have Dave Goski with the Markey Difference Maker of the Year Award.
I know I'm holding between you and the reception, so this is something that we like to do every year. Thank you for taking part in the 2017 Lewis A. Kelly Markey Difference Maker of the Year ceremony. As is tradition, I would like to recognize our former winners. As I announce your names, please stand. Marsha Ballard, Allison Yates, members of the Research Communications Office, including Jennifer Rogers, Donna Gilbreth, Heather Russell Simmons, and Kathy Anthony, Dr. Suzanne Arnold, Edna Ware, Carla Repass, Lisa Mink, and Mark Filbert. And I would like all of our difference makers in the past years to be recognized. I would also like to invite Dr. Lou Kelly, for whom the award is named, and Lisa and Mark, if Lou's still here, and Lisa and Mark to come up to the stage as we recognize our 2017 Markey Difference Makers of the Year. And as they're making their way up, I'll make my announcement of, due to a Academy Awards mix-up with PricewaterhouseCoopers between Moonlight and La La Land, we do not have envelopes this year, so I'm just going to hand Lou a piece of paper with the names of the winners.
Today brings us to our 6th year as we've been recognizing Markey Difference Makers of the Year. This year, these individuals were recognized as Markey Difference Makers of the Quarter for their efforts in clinical areas, thus making them eligible for the Markey Difference Maker of the Year award. Dana Stafford. Dana is a clinical nurse in our multidisciplinary clinic. Brittany Hatton. Brittany is a clinical service technician in the chemotherapy infusion. Jimmy Williams. Jimmy is a licensed practical nurse in our multidisciplinary clinic. And Robert Adams. Robert is a patient services coordinator senior in our Markey hematology program. Each of these individuals have gone above and beyond and have been recognized for their efforts by colleagues, faculties, parents, and/or family members of the patients that we work for every day.
These are the 4 individuals that are recognized as the Markey Difference Makers of the Quarters for their efforts in our research and administrative areas. Alicia Colliver. Alicia is a finance manager in the Center for Environmental and Systems Biochemistry. Susan Ruffett. Susan is a director of quality assurance and reporting for the Markey Affiliate Network. Robin Fisher. Robin is an administrative support associate for oncology services and integrative medicine. Cynthia Leadham. Cynthia is a clinical research associate for the Markey Clinical Research Office. All 4 of these individuals have given countless time and effort on behalf of the Markey Cancer Center, and we thank them for all that they've done.
What a privilege it is for me to see the fantastic work that's still going on at Markey. It's in good hands, and I'm delighted to see it moving forward. The Markey Difference Makers of the Year for this year, 2017, are Robert Adams, and Cynthia Leadham. Is Robert here? Cynthia?
And is our tradition, as you noticed earlier, we have something in a very similarly sized box that we like to recognize our difference makers of the year. So as they come up here, honor them for all the great work that they do every day.
In the vein of the Markey Difference Maker, you know you've built a wonderful cancer center when it is you want your loved ones to come here. My mother is being treated here at Markey, and she has nothing but the most amazing things to say. They live in an RV, so they can go anywhere, and they were in Texas. But here at Markey is where they choose to be in order to be treated, and I have to say it's the most amazing care that I've ever seen anyone given. So I'd like to have another round of applause for all those that work in the clinic. Now it's time for our door prizes, but we decided that we'd do this in a more pleasant atmosphere with food and wine present. So we'll convene to the president's room in order to distribute those. Thank you.